we'll just take that
Perfect.
clicker. Okay.
Fantastic.
Great.
Apparently I have to point it over there.
So welcome, everybody. It's my pleasure with Dr. Friedberg here to welcome you to the Spring Lymphoma Committee group meeting for SWOG. We're so excited to see so many of you in person. I know there's a lot of people on the Zoom as well, and glad that they could join us. It's really been fantastic to have those casual interactions that we all meet. We have a really busy agenda today. And we have both a mini symposium, as we've had in many years, as well as an update on some of our existing trials and a presentation of new trials that are going to be available to all of you, hopefully very soon. I'm going to go ahead and just say just a couple of really happy things, which is that yesterday, Dr. Friedberg in our closed session shared our accrual for the Lymphoma Committee. And just want to say that between 2017 and today, we are really the model for accrual. And this is really a testament to all the hard work that all of you have put into the trials, screening for patients, identifying them and getting them on. And so we're really grateful. And you'll hear some really exciting updates for example, the Hodgkin's trial and others, really showing where all the effort has been put. So anything you want to say?
No, it's great to see everybody, and really excited. Encourage interaction today too. There'll be opportunities for questions, and we purposely built that in. So please don't be shy, both in person and on the Zoom.
Yep, exactly. And for people on the Zoom, the chat is live, so you can definitely put things in the chat, which we can see, and then either read off or unmute you to be able to ask your question. And you can also use the raise hand function. So great. So it's really my pleasure to start our morning off with a mini symposium. And as all of you know, we're in a really exciting era and have been for some time, with the ability to do deep sequencing and understand more about the biology of the diseases that we are treating. With that in mind, our mini symposium today is novel diagnostic and therapeutic approaches to B-cell malignancies, and we have two outstanding presenters. I'm going to start by introducing our first presenter, Dr. Jesse SWOG, who is the CEO and the Chief Scientific Officer and co-founder of TwinStrand, which is a sequencing technology company that is used. I guess the best way that you explained it to me last night was that it's sort of like a chip in the machine. That is one way to think about it, but really excited to hear about some of the technology that has been developed. Dr. SWOG is trained at the University of Washington and Fred Hutch, and is still clinically active, which I think gives him an edge for a lot of the scientists and even for a lot of the companies that are out there in understanding what the real questions are. So with that in mind, we've asked him to talk to us about ultrasensitive duplex sequencing for applications in heme malignancies.
Wonderful. Thank you guys so much. So I run a company called TwinStrand. This is a University of Washington spinoff that I developed this technology at as I was a fellow. So I know many of the folks in the room and continue to work with there, and my clinical responsibilities are a part-time general oncology clinic at the VA. So I'm not a ultra lymphoma expert, but I certainly treat lymphoma patients. And so I'll talk to you today about things I think are particularly relevant to lymphoma, but it'll be a little bit more broad in terms of heme malignancy also. Fundamentally, I think most of you are probably familiar with the clinical applications of DNA sequencing in cancer and oncology in general, identifying certain variants that are present in a tumor at time of diagnosis and pairing patients with a therapy. And that's from a technical perspective, a relatively easy thing to do at this point. DNA sequencing is a new technology in the last decade, but at this point is quite established. When you have a tumor, a mass from a lymphoma, or a leukemia at time of diagnosis, and you sample that, the majority of the cells have a particular variant in it that's in the tumor. And that's a relatively easy thing to pick up, just like it's relatively easy to identify someone who has a germline mutation compared to someone who doesn't. But where the technology really breaks down is detecting a small amount of one thing that makes them a lot of something else. One lymphoma cell floating around in blood mixed in with 10,000 normal cells, or one, CLL cell, it's residual disease after a CAR T therapy. And so just kind of help you visualize this problem. On the bottom, I'm showing you a kind of, sequencing data generates just this massive computer files. And I've collapsed this down in a sort of a simplified way, where in the x-axis on that lower left, I'm showing you a position in the genome. And this is just the 400 base pairs out of the 6 billion nucleotides in the diploid human genome. And the y-axis is what's called the variant allele frequency, which basically means the percentage of molecules or cells you're sequencing that have a particular variant. Heterozygous or homozygous, meaning it's in one copy or both copies of something that's a germline, like it's inherited, is very easy to see. It stands out. It's 50% or 100% of molecules. Or at time of diagnosis, if you're looking in, take a biopsy of a DLBCL and you're looking for something and the tumor purity is mostly lymphoma, it's quite easy to see something that's in all of those cells. But if you zoom in and you look in the lower right and you make the y-axis show up at 1% as a maximum as opposed to 100%, you can see there's actually a lot of background noise. Every position looks like it has mutations, and those are technical errors. And so if one were trying to look for, say, residual disease, where you're looking for 1 out of 100, 1 out of 1,000, 1 out of 10,000, you could not see that. You couldn't detect that because the noise overwhelms the signal. Duplex sequencing is this technology, and I won't go into all the really too many details of how it works, but involves special biochemistry and special informatics that fit on both sides of kind of a normal standard DNA sequencer that are widely available and used, the same machines that are used for all the sequencing you ordered. And it improves accuracy by more than 10,000-fold. And so I'm showing you data here on the left is standard Illumina sequencing. The y-axis is, again, the variant allele frequency. And on the right is duplex sequencing of the same sample. This is actually from data from a CML patient 4 months prior to clinical relapse. And that one true mutation that you can see with duplex sequencing that was buried in the noise of the standard sequencing was I think this was a T315I mutation. So this patient was on, I think, yeah, whatever, whichever earlier, I think either imatinib or dasatinib, earlier generation TKI. And again, had I known that as an oncologist, theoretically, we might have been able to head off that, subsequent relapse by virtue of seeing that early drug-resistant mutation. The way the technology works is we're basically applying these unique molecular labels to every single DNA molecule. Of the bazillions of molecules you get when you extract DNA from a blood sample or a tissue sample, we put a unique fingerprint in every one, and then we make copies of it, and then we sequence those. And then we use the portion of the sequence read that has that unique randomized fingerprint to find everything that shares that, which means all those copies that share that particular pattern in the original top strand conformation or bottom strand conformation of these double-stranded DNA molecules came from the same original starting molecule. Any variation among those sequence reads must be a mistake. And so we're kind of recapitulating something that is used for maintaining high fidelity of DNA replication during cell division, kind of like looking for mismatches. And again, without getting into all the details, there are certain types of errors that are made in the sequencer that during the normal biochemistry for preparing things to go in a sequencer that we are able to identify and eliminate. Including mistakes that are made during the first round of amplification. And bottom line, at the end of this process, we get data out from the sequencer that then we process in the cloud and do this error correction, and we drop our error rate below 1 in 10 million. As a company, we're focused on this technology, and the product is literally chemistry at the front end and special analytic analysis software at the back end that you take your sample, you take your chemistry, you process it, you put it in a normal sequencer, and the data that comes off it gets loaded to the cloud where analysis happens. And we work with a ton of different companies, drug companies, regulators, academic groups. We are intimately involved in, I'll show you some SWOG data, literally related to AML though. Applying this tool in kind of broad applications where very low frequency variant detection matters. I'm showing you, this is from a Nature review paper I did a few years ago, kind of showing a lot of different applications for low frequency variant detection in across medicine. And the kind of 3 main ones we focus on as a technology company are, residual disease and cancer, something called genetic toxicology, which is, you know, won't talk about much today, and then allogeneic cell therapy tracking. And I'm going to kind of those two applications on the left, I'm going to show you, or on the right, I'm going to show you a couple examples of. Everybody here, you know, is familiar with minimal residual disease or measurable residual disease or molecular residual disease or meaningful residual disease, whatever you want that M to be. You know, bottom line, you know, in leukemias, this has been something that's recognized as highly relevant for a long time. We typically use flow cytometry. It's been less relevant in sort of indolent diseases, indolent lymphomas, until more recently, when we kind of knew people were never going to be completely cured. And now, as an endpoint, it's really valuable. How deeply do you put somebody into a or a remission? What kind of kinetics are you getting? So I'm going to show you some AML data, which again, sorry, it's not lymphoma, but it's highly related. We have a particular assay for AML that covers a portion of the genome that's mutated in about 95% of adult AML patients. I'm showing, this is sort of a analytical validation, so you can kind of, again, get a visual of how well this works. We took DNA from a young, healthy person who's 18-year-old male, so very low probability of CHIP. And we spiked in some very specific mutation, cell lines that had specific mutations. I'm just showing you a few of these, P53 and a couple of RAS genes, just a small subset of this panel. Top is normal sequencing. The middle is kind of a one level of error correction. The bottom is duplex error correction. The y-axis is zoomed into 0.1%, so 1 in 1,000. And so in the top with standard sequencing, there's a lot of noise. You can't see anything, you barely see 1 in 100. With the next level of error correction, you drop the errors a lot, but you still can't really get below about 1 in 500 to 1 in 1,000, because there are certain regions of the genome, for example, shown on the right, where there are false positives that have a locally high kind of error rate. And duplex sequencing in the bottom, you have no problem seeing down, in this case, 1 in 100,000. I had to make little inset plots to be able to see it in the same scale. So you get massively high specificity. And specificity is really, really important. You can have 100% sensitivity, but that doesn't help you unless your specificity is really good. So clinically, what do? You got great technical stuff, technical performance, who cares? Does this actually translate to something meaningful, actionable? This is from an old SWOG study, 0106. I think this was 7+3 versus 7+3 plus Mylotarg, I think. This was looking at day 28 marrow aspirates from, I think, this subset of maybe 80 or 90 patients, some who relapsed and some who didn't. The right side of the plot is kind of best in class Fred Hutch flow cytometry, looking at either relapse-free survival or time to relapse, and looking at the hazard ratio of those. It's around 2, kind of statistics in this case are okay, but kind of borderline. On the left side is duplex sequencing. This was done completely blinded, and we unblinded the data with our SWOG statistician, who said, hazard ratio of 5 to 7. That's pretty good. This was our first pass. We only had a little bit of DNA to work with. We hadn't done anything. We just created a specific cutoff, and the correlation with relapse-free survival, time to progression, and overall survival was phenomenal. So this is getting written up right now. And again, this is just showing you that, being able to look at this, a quite low level, really matters. You might look at this and say, well, like, look in that upper left one, you got some things that say duplex sequencing negative. Okay, they don't really relapse, but then, 20% of people are duplex sequencing positive, and they don't relapse out to 6 years. What's the deal with that? Is that, false positive? And the answer is no, probably not. This is day 28. We know that people relapse after day 28. That's why we do consolidation therapy. We wouldn't do consolidation therapy if people were cured. So these people underwent, subsequent consolidation, in some cases transplant. But it just shows that, people who achieve a level of residual disease below a certain cutoff at day 28, it's very prognostic. And so we, are looking at subsequent later time points, pre-transplant time points. I think we can make these distinguishing, even better. But it's again, the technology lets you see things that are quite special. In terms of residual disease monitoring, I think a lot of people are familiar with how we typically do residual disease monitoring in AML. In lymphomas, myeloid malignancies, including multiple myeloma, it's possible to use VDJ recombination, things like the clonoSEQ assay by Adaptive to track residual disease. But, one of the challenges is you're looking at, one locus, and occasionally you don't have rearrangements. So that assay is not, totally generalizable. With CML, we can follow BCR-ABL. And with AML, I showed you data from a panel that works quite nicely. But, for AML, most patients have mutations in a small subset of the genome, and many lymphomas and myeloma, it's much more genetically diverse. It's really hard to have like one assay that we can use, because the bigger your panel, the more patients it's relevant to, but the more kind of expensive the sequencing is. So how can you get around that? And then a second issue with lymphomas is, when you're measuring residual disease, where are you measuring? You're looking at marrow, or are you looking at cell-free DNA in the plasma? And if you're looking at the latter, there's just not that much of it around. So if you don't have a whole lot of material to work with, and you don't know exactly where to look, how can you go about actually, doing MRD monitoring in lymphoma? Well, one kind of basic statistic that it should be relevant from elementary school is, if you got a jar full of jelly beans, say 1 in 100 of them are red, the more jelly beans you pull out, the more statistically confident you are that that measured proportion is reflective of the true proportion. That's what a p-value is. So the more you sample, the more confident you are if your accuracy is really good. Our accuracy is, for all intents and purposes, pretty much perfect. We're very close to that. So we're limited by really sampling. But there's another element to this, which is we actually, when you're looking for residual disease, you don't actually care necessarily about the frequency of a specific mutation. What you really care about is the frequency of the clone, the leukemic clone or the lymphoma clone. And so the more deeply you sequence, the more cells you sample, the higher your sensitivity. But it also turns out that the more independent mutations you sequence that represent the clone, the higher your power. So what we can actually do is we can take a tumor sample and a normal sample at time of diagnosis. We can do a whole genome sequencing or whole exome sequencing on it. We can identify hundreds to tens of thousands of mutations that are patient specific, and we can develop a rapid turnaround custom assay using kind of newer forms of DNA synthesis, and get astronomically high accuracy, because we can track many mutations. So on the x-axis is how deeply you sequence. It's like how many jelly beans you pull out. The y-axis is the number of mutations or variants per cell that you're tracking. And you can see in the far upper right, if you sequence to 100,000 x depth, that's like think of that like flowing 100,000 cells, and you look at 100 mutations, you can achieve 1 in 1.5 million sensitivity. So if I don't have very much material, like for say I'm working with cell-free DNA, the way you can still get very high sensitivity, you're just tracking a lot more markers. And this approach works for anybody. Literally anybody. So it's you're basically making a customized assay per person. You've seen many companies starting to do this, and we're able to do this with about 2 to 3 orders of magnitude higher sensitivity. Last thing I want to say quickly is we can apply that same type of logic to something that is done by a lot of lymphoma docs, allogeneic cell therapies. We think about chimerisms. Anyone who does transplant, we think about chimerisms. What percentage of your blood or your marrow is from the donor versus recipient? Well, the same thing applies if you're infusing, say, CAR T cells. How long do they stand around? Where do they go? How deeply, do they disappear? And with allogeneic CAR T cells, which is something I think will be the future, off-the-shelf therapies, you have tens of thousands of polymorphic sites that are different between the donor and recipient. So we can take that same logic, and we have a single assay that will release officially later this year. We're working with a variety of allogeneic cell therapy companies on. We just track a bunch of polymorphic sites around the genome that can differentiate donor and recipient. This is doing spike-ins of from 1 in 300 down to below 1 in a million of an actual allogeneic cell therapy. This is from data that was from a phase 1 clinical trial that I believe is in front of the FDA right now, actually showing that when you spike this in and you do it in triplicate in a blinded way, we see exactly what we get, and spiked in 0, we truly get 0. So specificity and sensitivity of perfect down to below 1 in a million. This is some clinical data. This is from, again, can't disclose the company, but it's an allogeneic CD19 CAR. These are 2 patients that did very well long-term, and you can see 2 to 6 months out, you can see persistence at very low levels. The pharmacokinetics of this is quite interesting. In one case, subject 1, and within a few days dropped way down, in 3 weeks, it's down to close 1 in a million. Then there's an expansion event. So you can look in marrow, you can look in peripheral blood. You shouldn't see much in peripheral blood. Ideally, this should be in the lymphoma, it shouldn't be in the peripheral blood, but, being able to see what's going on. And the cool thing is literally the same analyte, whether it's blood or marrow aspirate, we can track both residual disease of the tumor and the pharmacokinetics of these drug products. This is data in the last slide I'm going to show you with Colleen Delaney, who's a faculty at the Hutch and also runs a company called Devara that does CD30, well, it's cord blood samples, where they basically take small cords that are not, eligible for transplant because the number of CD34 cells is too small, combine them and infuse them for a variety of applications. Initially this was for essentially trying to have sort of HLA mismatch transient hematopoiesis to kind of prevent, bad infections during that engraftment period. But actually, they found that actually seemed to reduce relapse risk in AML, presumably because of some kind of, graft versus tumor effect or immune, stimulation. This is looking, the FDA said this is great, but if you're mixing together, 8 or 10 cord populations and you infuse them, how do you know what's going on? Is one growing? Is one not growing? This is a blinded mixture from 16 different cords, made 8 different mixtures, and we were able to, deconvolve this mixture. So imagine you've got, even current clinical practice, someone has a cancer, and then you give them a double cord, and then the graft goes down, and then you give them another double cord. There could be 5, and then maybe you give them CARs at something. There could literally be 5 or 6 genomes floating around, and this technology lets us deconvolve those with very high levels of specificity and sensitivity. So we apply this again, MRD, tracking of cell therapies. We do a lot of stuff with CRISPR companies looking at off-target gene editing and sort of safety of those. Won't really talk about that, but in general, we focus on applications where this technology and the accuracy it brings, affects, can help affect change. It brings true meaningful information. I try to stay away from areas where standard technologies are perfectly good and really focus on things that are truly enabling. Love working with different folks. I'm a physician, I'm a scientist. I can speak your same language, and the type of collaborations that we're doing, academics and pharmacy is what pharma is what I want to keep doing. So happy to answer questions.
Question.
Yes, we have time for questions.
So really elegantly done, and I think highly relevant as we're planning trials moving forward. In fact, I think for every one of our ongoing trials, there's some assay for MRD or circulating tumor DNA that we're thinking of building in. I think one of the challenges in lymphoma that's certainly different from leukemia is that many patients get a needle biopsy that's about that big of large cell lymphoma, and we make treatment decisions and move along. We don't have the luxury of aliquots of bone marrow or blood that we can keep looking at. And how do you think this technology will fare knowing that limitation? That's actually, I think, been one of the big challenges in the lymphoma field of moving this forward as compared to our colleagues in the other blood cancers.
Yeah, so if we're going to be measuring residual disease by trying to do biopsies of a lymphoma mass somewhere, that's obviously not really practical. So these non-invasive samples looking at peripheral blood for rare circulating lymphoma samples, or marrow aspirates, rare lymphoma cells, or cell-free DNA. People should know what cell-free DNA is. I know it's more of a solid tumor thing, but it's basically cells that are rapidly turning over, dying, releasing these apoptotic fragments that have about a 2-hour half-life. They're at very low abundance, but that is really relevant, important information. So from the perspective of this tumor-informed residual disease monitoring, you only need the teeny tiniest little amount of material at time of diagnosis, and you could even often get that out of peripheral blood, and then use only these very, very easy to get materials. You can work with cerebral spinal fluid. You can work with urine. Urine probably wouldn't be that relevant for lymphoma, but for CNS lymphoma, it's a absolutely relevant thing.
So you think even with the initial biopsy, because you spoke to either getting, a lot of material or a small amount of material, but looking at a lot of different genes to define the profile. You think that with that small biopsy, even fixed, that you'd be able to do that with this type of technology?
With a small fixed biopsy, you would do like a kind of a standard normal accuracy sequencing, which is fine because it's a clonal population. And then from that, you know what's in the cancer. You have a peripheral blood sample that tells you what the person's normal genotype is, and you know the difference. And then using these much harder to work with samples, you apply duplex sequencing later to track, again, down to your 1 in a million level. So that's quite practical. And we actually deal with that with solid tumors all the time.
So I just have one more question. Sorry. Okay. So this was really beautiful. Thank you so much for speaking today. And I'm sorry that we asked you to cram so much into a short period of time, because this is really exciting work. I know you have a lot of different, stories, if you will, that your company has been working on. Where does like epigenetic modification come in? Because I think in lymphomas that actually might be very, maybe even, I don't know if it's more relevant, but it's very relevant.
Yeah, epigenetics is a super important thing. And I think there's really two ways of thinking about sort of two different things. One is if you have a lymphoma at time of diagnosis, how do you look at all the things about it? It's epigenetics, it's sequence, it's proteomics, it's whatever that helps you predict how it's going to behave long-term. Residual disease monitoring is saying, I don't care about saying, oh, there's a inversion here or a high expression here. We're empirically measuring what actually happens. And so if you don't know all the markers to look at, you're able to, again, empirically measure what's happening with the tumor. And that will give you a lot better ability to go back and correlate it with things like epigenetics and other things at time of diagnosis. Ideally, you want to be able to take time of diagnosis and predict what's going to happen. The way you do that is with correlative studies. So I view this as one important tool, but it's absolutely not the only tool. And all those other things are important. And we do a lot of joint collaborative studies with other groups and other technologies, but we're really, really good at saying how much of the disease is left, how fast does it go down, what is the granular response to a particular therapy. That's our sweet spot. Yeah.
Thank you so much. Really appreciate it. Oh, sorry.
Sorry, could I ask you a question?
Yeah.
Sorry, great talk. Thank you. One, do you even need that initial specimen, or can you do pretty much the same thing on blood even from the beginning?
You probably could do the same thing on blood. If there's a reasonably high level of disease, you could get it out of blood in kind of a standard liquid biopsy. It's ideal to know what the cancer is compared to normal, because the problem is there are a lot of, if you just sequence your residual disease later and see a little bit of something, how do you know that wasn't a chip clone? How do you know that wasn't a somatic mosaicism? There are a lot of inherited things that if you look in a cancer and you don't look in normal, there are inherited variants that look pathogenic, but they're literally inherited. And so that's why it's nice, but you could do it without a biopsy.
Great. My second question is, this is obviously an area of very active investigation, and there are other groups working on trying to improve the signal to noise ratio. Can you compare perhaps how your technology is different from some other companies that are doing some of the work, the Stanford group or let's say the Cornell group?
Yeah. One of the unique things about us is a lot of it is the technology and the IP and stuff. I think tell you a couple of things. One is that you guys probably know Foundation Medicine, and their liquid biopsy product is twin strand technology inside. Guardant Health recently got sued by us for using our technology. And the group, Ash's group at Stanford, the CAPSeq is a duplex sequencing. They sold that company to Roche, which owns Foundation Medicine, which is now using our technology. So I'm a scientist and physician. I'm not like just a wheeling and dealing business guy, but we have good technology. It's sort of protected, and this company that's University of Washington, and again, we kind of have like a little bit extra clinical knowledge and experience. It's a little bit different than some of the companies that are pure technology. There are other companies doing various things. The ones that are doing something else, it's kind of that middle level of accuracy I showed you, which is not as good. Some applications is perfectly fine, but I think we're pretty good. Yeah.
Thank you so much.
Last question. Go ahead.
Well, I have two questions, but the first one has already been asked about how this compared to other assays. The other question is, so when it comes to lymphoma and cell-free DNA sequencing, assays have been quite successful in aggressive B-cell lymphoma, but not as much indolent B-cell lymphoma. And it remains unclear whether it's due to tumor biology or tumor burden. And so just by increasing sensitivity, you may overcome that. How does duplex sequencing perform in indolent B-cell lymphoma, if you have any data?
Let's see. I mean, I guess, what do I have in indolent? B-cell lymphoma. You know, from a technical perspective, it's all the same. We've done certainly some stuff in, we've done CLL, okay, and we've looked for BTK inhibitor, pre-existing resistant mutations to throw out. But I guess I don't have a large, clinical data set specifically in indolent lymphomas, but we've done things in solid tumors, colon, breast, lung. We did a lot for AML. We're, I don't know, you guys know Rich Little. I don't know if he's on the call, but
Should be on.
okay, the MyeloMatch program, which is AML, CTEP's large kind of match focused purely on myeloid malignancies. We're doing all the MRD assessment for that. So I would love to get in and have a large dedicated study and all the things you just said, but I just need some partners. So email me.
Will do.
Yeah.
Thank you.
All right.
Great. Thank you so
Thanks.
much. So thanks so much. What a great way to start the morning. I'm going to move on to our second speaker in our mini symposium, who is one of our own within our SWOG Lymphoma Committee, Dr. Brian Till. He is an associate professor at the University of Washington and Fred Hutch Cancer Center. And he's been an integral part of our lymphoma committee and has a career really focused on immunotherapy, most recently augmenting cellular therapy in lymphomas. So we've asked him to talk about some of his lab work here today. Brian?
Thanks, Sony. Yeah, so thanks so much, Jonathan and Sony, for the invitation to speak. It's really a pleasure and an honor to be able to present our data to this group. I do have a relevant disclosure, which is that I have a patent for the CD20 CAR that I'm going to be talking about today, licensed to Mustang Bio, who's also funding the trial. And I'm also going to be speaking about non-FDA approved treatment. So I don't need to go too much into detail about how CD19 CAR T cells work. I think we all know they're pretty effective, but there is room for improvement. At least in large cell lymphoma, only less than half of patients get a durable complete remission. For follicular mantle cell lymphoma, we kind of need longer follow-up to see if there's a plateau or what happens there, but there's certainly plenty of patients who patients who progress. CD19 loss isn't the main reason for treatment failure, but it does contribute. There are a certain number of cases that do lose CD19, so it makes sense to target alternative antigens. Some of those, there's a lot that are being looked at. There's CD20, which I'll only be focusing on today. CD22, kappa light chain, ROR1. So there's a number of them out there. You could envision these being given either in combination with CD19 CARs or sequentially, or there may be some that even work better. So CD20 is a pretty obvious target. It's expressed on the vast majority of B-cell lymphomas, just like CD19. It's stable on the cell surface. It doesn't really get shed or internalized with antibody binding, unlike CD19, which gets internalized with antibodies at least. There's no decoy soluble antigen in the serum. And it's got 2 decades or more of use of proven track record as a target for immunotherapy with rituximab and other CD20 targeted agents. And despite the selective pressure of multiple cycles of therapy that everybody goes through with rituximab, loss variants do occur, but they're relatively rare. It's CD20 remarkably preserved over time in the disease course. So I'm going to spend most of the talk today kind of focusing on the CD20 CAR that we've developed in my lab, but I'm going to kind of give a little bit of an overview of the landscape of some of the other CD20 involved CARs out there. Turns out most of them are bispecific for CD19 and CD20. So here's one from the Medical College of Wisconsin, where they treated 17 patients with a dual domain of CD20 and CD19 together on the same CAR construct. They have a 4-1BB co-stimulatory domain, and they treated 17 patients, about half of them with large cell lymphoma, most of the rest of them with mantle cell. And they had what I would consider to be fairly comparable, very good response rates and a good CR rate of 64%. I would say what you might expect, it would be similarly in line with other commercial CD19 CARs for that mix of histologies. And similarly, I would say a similar safety profile. They did another study where they used the same construct, but they cultured the cells in IL-7 and IL-15 to try to enrich for a more memory and naive cell phenotype. They treated 22 patients, had 11, half large cell, most of the rest mantle cell lymphoma. And again, similar response rates and similar toxicity to their previous study and to what's seen in the CD19 CAR commercial space. So not necessarily obvious that the bar was raised there, but certainly another promising treatment to add to the arsenal. This is a little bit of a larger study from China that also used a CD19 CD20 bispecific CAR. I think it was also a 4-1BB. And this was a mostly large cell population of various forms of large cell lymphoma. There were some folliculars in there as well. They interestingly used liposomal doxorubicin as part of their lymphodepletion regimen, but they had impressive CR rate of 70%. Overall response rate was 78%, and similar again safety to what we've been seeing with most of the CD19 CARs. This time, with fairly decent follow-up, they have what looks like maybe a plateau of around 50% or maybe even a bit higher. So there's a suggestion that that may have actually contributed modestly, improved modestly over the commercial CAR, which we usually see about 40% plateau for most of those. So that one will be one to watch. And then finally, there's this construct from UCLA that the little twist that's a little different from them is that they use a CD62L selection for their T cells to sort of enrich in naive and memory T cells. They used a standard lymphodepletion and treated 5 patients with a mix of histologies, mostly follicular, and saw responses, actually complete responses in 4 out of the 5 patients. All 5 of them got CRS, but it was all grade 1 and there was no neurological toxicity. So that also looks pretty promising, but still pretty early days for that. So I'm going to shift and really just spend the rest of the talk really talking about the CAR that we've developed at Fred Hutch. I go back to 2006 working on in the CAR T cell field when I joined Ali Pressa's lab. So he had set up a collaboration with Mike Jensen from the City of Hope, who had developed a CD20 CAR. And so I kind of took over when I was a fellow, the clinical trial that he had set up, which ended up being the first published CAR T cell trial for lymphoma. And you haven't heard of it because it didn't work very well. We used a naked DNA plasmid to modify the cells, and it just wasn't a very effective way to manufacture the cells. So we went back to the lab, and when I kind of got into my own lab, we set up several years of optimizing different forms of different modifying of the CAR constructs. We looked at different antigen binding domains, took the mouse one and changed it to a fully human, and we looked at different spacer domains and we looked at different co-stimulatory domains, and basically came down to those top 2 on the top there, where it was a CD28 only versus a 3rd generation that was CD28 and 4-1BB. In the lab, they pretty much worked almost identically. We ended up going with the 3rd generation one, just partly because it was different than what everybody else was doing, but also because of a theoretical benefit of adding that 4-1BB domain. So that's the one we ended up deciding to take forward. And we did compare it in a head-to-head mouse experiment with a Juno CAR, and saw that it compared favorably, at least in this one small mouse experiment. So that was encouraging, and similar persistence in the mice as well. So this is the construct that we're using. It's again a 3rd generation, fully human CD20 targeted CAR. It's got a longer, so for CD20, you have to it's a pretty membrane proximal antigen. So you have to have a long spacer to get the right space between the target cell and the T cell. And so we use the IgG1 FC region, and we learned that you have to put mutations in that so it ablates FC receptor binding. And then you get, we had a CD28 and 4-BB domains. There's also this truncated CD19 selection marker that is handy if you want to track these cells in vivo. It's not so great if you want to treat patients who have already had a CD19 CAR. So we're working on getting that, because it targets, now puts the target on the back of our CAR T cells that can get destroyed by those any residual CD19 CAR cells. So eventually we are going to be getting rid of that, but we're sort of stuck with it for now. So here's the clinical trial that we're running. SWOG Zone Maziar Shadman is the clinical PI of the study and doing a great job. So this is a single institution phase 1/2 study, mostly phase 1. And it enrolls pretty much any histology of CD20 positive relapsed relapsed lymphoma. Large cell lymphomas have to have 2 prior lines of therapy. Mantle cell needs to have a prior BTK inhibitor. CLL has to have a prior BTK and venetoclax failure. And because of that CD19 truncated marker that we have, if they've had prior CD19 therapy, we have to demonstrate that they've had recovery of their normal B cells to show that there aren't functional persisting CAR T cells that are left, which is a kind of a hard bar to, as you might imagine, these patients have gotten a lot of B cell therapies to have that happen. But otherwise, it's a pretty standard trial. Everybody gets like cyclophosphamide and fludarabine. There's about a 10-day manufacturing period. Then the cyclophosphamide fludarabine, 2 or 3 days later, we get the T cells. And at the dose levels shown there, it's in an escalating fashion. So we have had this going on since actually 2017. And when we first started, we used a different manufacturing process where it was similar to the Juno process where CD4 cells and CD8 cells were grown in parallel and then combined at the end in a 1-to-1 ratio. There was also a restimulation step to kind of boost CAR expression and CAR and growth as well, but also kind of drives them towards more of an effector or maybe even exhausted kind of phenotype. And we treated 7 patients with that process. And another caveat was that we also tried to get away with just cyclophosphamide alone in the first 4 patients before we bit the bullet and combined with fludarabine. But overall, it was not successful. We didn't see any responses in those first 7 patients. The expansion wasn't very good. And we're actually having trouble manufacturing enough cells. So we ended up putting the study on hold, revamped the manufacturing process to a process where we combine the CD4 and CD8 cells at the beginning of cell culture, eliminated the restimulation step. So it's a single stream, shorter, went from 15 days to 9 days, a much shorter process. And it actually ended up not only making the CD8 cells expand better, but also there's actually a whole paper we're working on now talking about the different effects on CD8 function that this had, where the CD8 cells have a less differentiated phenotype and have a better function. And so that's the manufacturing process we switched to. So we've now treated 20 patients as of the ASH data cutoff, which is what I'm going to use for this presentation, and just focus on those 20 patients. So here are the patients. 15 of them are follicular, so that's kind of what we focused on, mostly due to the competing, commercial CARs that were available for other histologies. And these are high-risk follicular lymphoma patients. 2/3 of them were POD24. They had a median of 5 prior lines of therapy, and a few of them had prior large cell transformation that had kind of relapsed back to follicular. Then a smattering of other histologies, a couple of mantle cells, one CLL that was pretty heavily pretreated, a CD19 negative large cell lymphoma, and then a Waldenstrom's who had 10 prior lines of therapy. So the response rates are shown here, and kind of highlighting the follicular lymphoma, where we saw 93% response rate, 73% complete response rate. We basically only had 1 patient who didn't respond. And when we first looked at her PET scan at day 28, we actually thought, Maziar and I thought that she had a CR because all the tumor was gone, but the radiologist picked up a subtle new little lymph node that turned out to be real. It was biopsied and it was transformed to large cell. It was still CD20 positive. The 2 mantle cells both had partial remissions that were short-lived. The CLL and large cell lymphomas both had durable CRs so far, and the Waldenstrom's patient had a very good PR that was it cleared the marrow, but there were still a few spots lighting up on the PET scan that were trending towards a CR over time, but as of the data cutoff was still a PR. There's a suggestion that at the higher dose levels that we've gotten to, the response rate may be kind of even a bit better. And the durability is shown here. Most of the patients are still in remission. There were a couple of follicular lymphoma patients that had relapsed at around 9 months with CD20 positive disease. There was 1 patient with a PR who relapsed with CD20 negative disease. That's our only antigen loss relapse so far. But all the other patients, and then the 2 short-lived mantle cell lymphoma remissions, but everybody else is still in remission so far. So far that I would say that the efficacy and the durability of responses is, again, kind of in line with what you would expect for like axi-cell, for example, but I think maybe where we have some differentiation is when it comes to safety. So the safety profile has been really quite encouraging. The CRS rate of all the patients, all 20 patients, was 40%. Which is not that different from Tisa-cel or Lysa-cel. However, it's all grade 1 and 2, and we haven't seen any grade 3 or 4. The neurologic toxicity is only 10%, and it's all grade 1 and 2. If you look just at the follicular lymphomas off to the left side, the CRS rate is only 33%, mostly grade 1, and no neurological toxicity at all. We did see one follicular lymphoma patient who had MDS, but this was a very heavily pretreated patient with multiple treatments you can see there, including radioimmunotherapy and Revlimid. So we attributed it to that. And then just, cross-trial comparisons are always fraught, but just to give you some idea of how this stacks up, if you focus just on our follicular cohort and compare that to the Zuma-5 with Axi-cel and Elara here, you can see with Axi-cel, it's a pretty dramatic difference. Tisa-cel, it's a little more subtle. It's on the surface is fairly similar, although it's a little bit apples and oranges because they use the Lee 2014 criteria for CRS and CTCAE version 4 for neurotoxicity, which and compared to we use the ASTCT criteria, which are a bit more stringent on most of the parameters. For example, with the Lee criteria, you can be on a pressor in the ICU and still have grade 2 CRS, whereas that's grade 3 with ASTCT. You can have a grade 1, you can have a seizure that's grade 1 with CTCAE, and that's also grade 3 for ASTCT. So we didn't see anybody there was nobody in the ICU. And you can also get a sense for the tocilizumab usage that it's pretty low, and it's just a less toxic product, so it seems. So this is just a quick slide to show that if you compare our old manufacturing to the new, we really see a pretty dramatic difference in how, at least 2 log of difference of how well the cells expand in vitro. And we have seen pretty robust expansion overall. The cells have also persisted out pretty well out to up to 2 years so far in some of the patients, and with a preferential expansion of CD8 over CD4. So the data were promising enough that our industry sponsor or our industry partner, Mustang, has decided to open their own multi-center trial. They're moving the manufacturing from, instead of the Hutch, they're going to have their own manufacturing facility. There are a few tweaks to their process that are slightly different. And so from a FDA perspective, it's a different product and they have to start over at phase 1, but it's a fairly short phase 1 run in. There's 3 different cohorts that are going to be independently dose escalating. They're starting off at what I think is a somewhat of an aggressive starting dose of these different ones. They're starting at dose level 3 for the follicular cohort, and what's our dose level 2 for their the other cohorts. And then the phase 2 is going to have basically similar arms with a large cell lymphoma arm, a follicular arm, a CLL arm. Then there's sort of this basket arm of all the other histologies. So this is going to be opening very soon, or maybe even this month, or hopefully by next month. Multiple sites. I think Pat Reagan is going to be at one of the site PIs that we're excited to have Rochester on board, and so we'll see how this goes. So just in summary, CD20 I think is a reasonable target to target for CAR T cells, and the MB106 that we're working on has shown promising safety profile and efficacy so far, particularly in follicular lymphoma. So we're interested to get some other histologies in there and see how it fares. Some early promising signals in CLL and Waldenstrom's, but just with like a single patient basically. So and the study is opening. So I'll stop there and just acknowledge my former mentor, Ollie, the late great Ollie Press, who got this all started. And Mazir Shadman is the clinical PI, who's also going to be the national study chair of the multi-center study as well. So thank you for your attention, and any questions we might have time for a couple.
Yeah. So that was really great. And it's really exciting to see how, you've developed your career even very, very early in this whole field. It's just exploded. You started out by mentioning the bispecifics. So, maybe this is a little bit parallel to what you presented, but with the bispecifics that are being developed, how do you see, this type of approach fitting in with just bispecific antibodies as opposed to CAR?
Oh, bispecific antibodies. So do you mean the bispecific CARs or the bispecific
I'm sorry,
antibodies?
the bispecific CARs.
Yeah, the bispecific CARs. Yeah, it's a good question. I think it may just come down to ultimately just the data of, safety and efficacy of, I think from when I look at the data, it looks like I can't really distinguish very much from what I've seen of those studies. Of if you compare just CD19 CARs versus the bispecifics, they look to me almost identical, except for maybe that Chinese study where there was a little bit of an increase in the PFS. Yeah, they haven't really described in terms of antigen loss. They do seem to have. There aren't many patients who lose both antigens. So you could argue from like just an antigen loss standpoint, it may be just as good as the CD20. So yeah, it's a hard point to argue with, other than to say like if that gets chosen over a CD19 CAR, maybe if that could replace both, arguably maybe. But if it is functioning similar to CD19 CARs and then somebody relapses after a CD19 CAR, is there an advantage of reaching for a bispecific over just a pure CD20? Hard to argue maybe that is the case. But again, I think it'll probably come down to just where do the data look the best maybe, and that's what people choose.
This may be a question as much for Maziar as for you, but so I thought very interesting and compelling data, and the safety is obviously a big deal, and that may end up being the answer to my question. But with 3 established commercial products now that have gone through rigorous and confirmatory testing with randomized trials, what is sort of the development vision for something like this? How are you going to ultimately either displace or add to the current therapeutic armamentarium?
Yeah, it's a good question. I'll be curious to hear how people like Pat and you, if you put patients on the study, what you say to the patient in the room. I think when we've consented patients, I think we have enough where you can see from our pattern of enrollment that we really didn't feel comfortable telling a patient you should get this over, Lysocell, because it's, we've treated one patient with large cell lymphoma. So it's kind of hard to make that case. But when you've had 15 patients with follicular lymphoma and have seen the pattern there, it's enough data that I think we've felt comfortable saying you could get Axi-cell. Here's the data for Axi-cell. Here's what we've seen. And a lot of patients have actually, being presented with both of the data, have chosen this trial. So, but I think it's still going to be tough, maybe a tough sell for some of those other histologies. And, I don't know if there's other investigators at other centers that are more comfortable, potentially having a patient try out this thing. I think it's going to I suspect there's going to be a lot of CD19, post CD19 failures that are going to be enriched. That's probably for a lot of investigators. That's kind of the attractive part of this trial, but I'm hopeful that we can sneak in some other, more CAR naive patients as well. And then there's going to be, I think, we're also interested in some of the more orphan types of histologies like Waldenstrom's, and we've just expanded our accrual to include CNS, primary and secondary CNS lymphomas. So there may be some kind of niches outside of the main histologies as well. So I have a bunch of backup slides that we can have to probably push through here. Sorry, get to your next. There you go.
That's great. Thanks so much.
Let's give both of these speakers another.
So that was really fantastic. And we're going to move on to the second part of this morning, which is new concepts. We have 4 new concepts to present. Very excited to hear about each of them. 2 of them are a little bit further along, and we're going to start with those 2. So first, I think presenting virtually, we have Dr. Brian Hess from University of South Carolina. And his presentation will be on S2114, randomized phase 2 trial of consolidation therapy following CD19 CAR T-cell treatment, very appropriate for the talk we just heard, for relapsed refractory diffuse large B-cell lymphoma or grade 3 follicular lymphoma.
Great, thanks, Sonali. Yeah, I think it's a great way to follow those last 2 talks, especially that MRD talk that started off the meeting. So thanks for asking us to present this update today. The study is moving along nicely. We just submitted the protocol to CTEP a few weeks ago, and happy to give an update. So next slide.
Sorry.
So we've had a lot of contributions, both from within SWOG and outside of SWOG, and It's been a great effort from everybody involved, a great group effort, and we've really appreciated that so far. Next slide.
Sorry, I'm hitting the red button. Yeah. There we go.
Great.
There's a bit of a lag.
Okay. So I wanted to give a quick background that kind of frames the question of what we're asking in this trial, but I'll try and go through it quickly. The three columns on the right are the landmark trials that led to the approval of axi-cel, TISA-cel, and lyso-cel, respectively. You see the best overall response rates ranging from 52 to 83%, and the CR rates from 40 to 58%. And it's important to understand that these are best overall response rates, meaning that patients did have stable disease or PR on their first assessment after CAR T infusion, then converted into CR. Next slide, please. And then this is looking at these studies again, basically showing that patients who achieve a complete remission do far better than patients who achieve a partial response or stable disease as their best response. And also, it looks at like patients who have a complete remission at 6 months or longer. The majority of these patients, the greater majority of these patients are staying in a remission going forward. Next slide. I would also be happy to share my screen if that's easier. I have these slides up, Jonathan.
It's apparently better this way, we've been told.
Okay.
But I'm not sure why this clicker isn't working. I can stand if it's better.
Great. Okay, so these are two real-world studies looking at outcomes based on the day 30 PET. And you can see that patients in CR, both of these studies were axi-cel for DLBCL. And patients in CR at day 30, the majority of them are still in CR at day 90. And about a third of patients in both of these studies converted from partial remission at day 30 to CR at day 90. And patients with stable disease, a minority of these patients were found to have stable disease at day 30. And a very small proportion of them are converting to CR at day 90. Next slide. And of course, this is the update that we all saw at ASH this year, where CAR T-cell therapy was compared to salvage chemotherapy and autologous stem cell transplant as second-line therapy. And with the primary endpoint of all these studies being event-free survival, there were some nuances in terms of how they define that in these studies, but you can see that two of these showed a statistically significant improvement in event-free survival compared to standard of care, the ZUMA-7 trial and the TRANSFORM trial. And also interesting in this trial, you see that the CR patients are, as the best response, get a bigger piece of the pie. Almost 2/3 of patients have their best overall response graded as a CR. What we don't have in these trials, at least right now, is how many patients had stable disease or partial remission at the first imaging after CAR T-cell therapy and then converted to CR. That did happen a reasonable amount in one of these trials, but it's not public right now, so I won't share. But that did happen fairly enough to discuss in one of these trials. And also interesting, even that though patients, more patients had a CR in these trials, such as ZUMA-7, the 24-month event-free survival in this trial, only 41%, highlighting that most of these patients will relapse. Next slide. So this is just a summary looking at outcomes based on first response for CD19 CAR in DLBCL. We expect that about 50% of patients will have a complete remission at that day 30 scan, and the majority of these patients will go on to have a durable remission after this, although there are a good amount of patients that we expect to progress after that day 30 CR. About 15 to 20% of patients will have progressive disease either at that day 30 scan or prior to that. And we know that these patients have a poor prognosis. And then about a third of patients will have partial remission or stable disease at that day 30 scan. And a minority of these patients we're expecting to convert to CR. And we are expecting the majority of these patients to relapse. So this is really the patient population that we're identifying as high risk for relapse, that we can potentially institute consolidation therapy for these patients and the patient population that we're identifying in this trial. Next slide. And again, just trying to identify patients that are at high risk for relapse and predicting durable response. Minimal residual disease in this space has been shown to do that in multiple platforms, including CAPSEQ. And this is a big part of our trial, and it's the biggest correlative aspect of our trial, where we're going to be monitoring minimal residual disease on patients who have stable disease or partial remission at day 30, but also who have CR at day 30, again, to try and evaluate this high-risk population or refine this high-risk population. Next slide. And patients that progress after CAR T-cell therapy, they don't do as well in terms of responding to other therapies in terms of overall response rate. And even if they have an overall response, the response tends to be very short-lived. Next slide. There are a few novel therapies that have come up in this space that are more promising. And I'm highlighting a few that we're going to be utilizing in this trial for consolidation therapy. So that first column with mosunetuzumab, in terms of overall response rate and CR rate, this is a summary from the plenary in 2019 at ASH, where amongst 124 patients with aggressive lymphoma, the overall response rate was 37%, CR rate 20%, and very similar in patients who had prior CAR T-cell therapy. And I think one of the most interesting parts of this was looking at the patients that had CR. 70% of them were still in an ongoing CR at 16 months. About 30% of them had cytokine release syndrome. Only 1% was grade 3 or worse. Also similar in patients with prior CAR T. And ICANS had a very low incidence. They did report neurotoxicity, which had a higher incidence, but when they graded ICANS by ASTCT, it was very low in this trial. I put polatuzumab up here because we're utilizing it in this trial. That's from the Romulus study, but that was given, that was with this agent given with rituximab, and at a dose of 2.4 milligrams per meter squared, we'll be using 1.8 milligrams per meter squared. And then the last column is the combination of mosun and pola, and an update from ASH this year, and Manali might touch on this later in the meeting, but some very promising response rates, and especially CR rates in aggressive lymphoma, where the CR rate is close to 50%. And again, relatively small numbers of 11 patients, but very promising, with 2/3 of patients responding and close to 50% with the CR rate. And 18% of patients had had CRS, most of them being grade 1 and no grade 3, 4. Next step, or next slide, please. So obviously, CAR T-cell therapy in this space has been a huge breakthrough for these patients that otherwise didn't have other reliable therapies to lead to a durable response, but the majority of patients are still relapsing. Minimal residual disease has been shown to reliably predict relapse in these patients. Those that do relapse have a minimal response to other therapies, and their prognosis is poor. The best outcomes are definitely in patients who are able to achieve a CR. And we expect that the majority of patients with partial remission and stable disease will progress. And so this is the patient population that we're identifying in this trial, trying to institute consolidation therapies to them before they relapse. Next slide. So this is an updated schema, and Jonathan, you can click a couple more times for time's sake, because there will be a couple things popping up. And one more, and that's good. Okay, so the updated schema, again, we're identifying patients who have relapsed refractory DLBCL or follicular lymphoma grade 3B who are candidates for commercial CAR T. There will be 2 registrations for this trial. The first registration is taking patients that are candidates for commercial CAR T, and we'll be attempting to enroll close to 400 patients to first step registration. All patients will have a day 30 PET scan, and that will be done between day 25 and day 40, and they will all be centrally reviewed. For the patients that have progressive disease, those patients will move on and just have physician-directed treatment for their progressive disease, whether it was at day 30 or before. For the patients that have CR, they'll continue on trial to be observed, and they'll have we'll have dedicated response assessments or scheduled response assessments, and have biomarker draws at these response assessments, again, to help evaluate minimal residual disease in this population, to identify a subset that would potentially be relapsing and when they would be relapsing. For the patients with stable disease or partial remission by central review, these would be the patients that would be eligible for step 2 randomization and be randomized to mosunetuzumab alone, polatuzumab alone, the combination, or observation. It'll be in a 1-to-1-to-1 randomization. We estimated that the observation arm would have a 1-year PFS of 25%. And so we'll be comparing each of the intervention arms to observation. And our goal is that the 1-year progression-free survival improves to 50% or increase of 25% in all of these arms compared to observation. And also with the observation arm, we are allowing patients to cross over to receive the combination of mosun and pola if they progress within 12 months of receiving CAR T-cell therapy. And I'll go over some of the parameters for that in a future slide. Next slide. So the primary objective, again, is PFS at 1 year for these patients with stable disease and partial remission at that day 30 PET, which is, again, centrally reviewed. And each one of these intervention arms is compared to the observation arm. We'll also be looking at secondary objectives, overall survival, complete remission conversion rate, adverse events. We'll be looking at the crossover arm, a number of parameters within that. The most important biomarker objectives that we're looking at have to do with total metabolic tumor volume at day 30, as well as minimal residual disease, which I mentioned, which we'll be looking at in patients not only with stable disease and partial remission at day 30, but CR at day 30 as well, and looking at that going forward. Next slide. So important points about first step registration. Again, you have to be a candidate for commercial CAR T, have DLBCL, follicular lymphoma grade 3B, or primary mediastinal. You can have transformation from follicular lymphoma or marginal zone lymphoma, but you can have CLL, a Richter's transformation. Bridging is allowed, but you can't have pola or mosun within bridging. So something like BR pola is not allowed for bridging in this study. You are allowed to have received pola previously, which is important because many of us saw in ASH, again, the data of pola with cytotoxic chemotherapy in frontline, and expect that to be approved soon. So if you've received a regimen containing pola, such as pola RCHP, as long as you're not refractory or progress within 6 months, you'll be able to enroll on the first registration. And then if you've received bridging, a repeat PET is mandated before starting lymphodepleting chemotherapy. Key exclusion being non-commercial CAR T, previous CAR T, non-conforming CAR T products. This happens in a minority of patients, but it happens actually fairly regularly. I'm sure a lot of us have had products such as this. So if the patient's receiving the product on expanded access or a single patient IND, they would not be eligible. And they can't have any active CNS disease. So step 2 registration, again, stable disease or PR by central review at day 30. You can register up to day 60 after CAR T cell therapy, which is important because we have criteria regarding bone marrow function. The ANC needs to be 1,000 or greater, platelets need to be 75,000 or greater in order to enroll. We see that a lot at patients at day 30, and we see that improve over time. So extending that window to day 60 hopefully allows for more patients to be enrolled. No CRS within 7 days of registration, and no ICANS greater than grade 1. In terms of stratification, we're stratifying stable disease versus PR. As we've shown, the minority of patients have, a small minority of patients have stable disease at this day 30 PET scan, but in pretty much all the studies that are reporting this, they do much worse in terms of their relapse rates compared to PR. And then in anticipation of the recent approval that we saw last week of CAR T and second line therapy, patients will be stratified as receiving CAR second line versus third line or greater. Next slide. So next slide is third step registration once we get there. So this is going to be for a crossover. Again, patients are eligible to cross over to receive the Mosin-Pola combination if they progress within 12 months of CAR T infusion. They must have imaging that clearly demonstrates that they have progressed compared to the day 30 scan. They will have scheduled response assessments, and they can get scans outside of those response assessments, but they have to have symptoms that are concerning for progression to justify that. Much of the clinical laboratory criteria is consistent with step 2. You must be registered within 28 days of progression and can't have any lymphoma directed therapy prior. Next slide. So just a couple more slides here. This is the treatment schedule for all 4 arms for the consolidation for patients randomized to step 2. You can see the step-up dosing for mosunetuzumab for arm 1 and the combination arm in arm 3. Polatuzumab in arm 3 will start to be instituted in cycle 2, day 1, and all of these cycles are 21-day cycles. Next slide. And so this is just showing how the response assessments and biomarker draws are going to be scheduled for the patients who are randomized to step 2. So we're not basing this on days from CAR T cell infusion. We're basing it on days from randomization. So obviously everybody has a pre-lymphodepleting scan, PET scan, and a day 30 scan. And then once they're randomized, they will have their next response assessment 60 days from randomization. And we're encouraging this to be done prior to cycle 4, day 1. Also, if you, after this day 60 scan, if you're in a CR, you can get CT scans if it's denied by insurance. Next slide. So for the patients that have a complete remission at day 30 or have stable disease or PR at day 30, but are not a candidate for step 2 registration for one reason or another, maybe cytopenias or other toxicity, the schedule of assessments and biomarker draws is going to be based on when they received CAR T cell therapy. So a little bit different. But again, this is the most important group in this group, and probably the most important, the largest group, likely the largest group registered to step 1 are these complete remission patients, who make up about 50% likely of patients at that day 30 PET. And we'll be evaluating them with response assessments and MRD sampling within 72 hours of those response assessments with these parameters. Next slide. So again, we've had a ton of help from other groups in getting this project moving along. We've had a lot of working groups and a lot of contributions from everybody, including BMT-CTN, who presented this at the State of Science Symposium last year, which is, and it was viewed very favorably. This trial was presented to Lymphoma Steering Committee last fall and received approval. We were working on the protocol and just submitted to CTEP last month and are likely going to get comments back in the next couple of weeks. We anticipate study activation sometime this summer and very hopeful for that. So this trial is trying to determine a safe and effective regimen for consolidation therapy for these patients. If we're able to find that regimen or multiple regimens, then we'll likely move forward with a phase 3 study and possibly an MRD adaptive approach, depending on what our correlative studies show. And even if we don't find a safe or effective regimen, hopefully we'll gain a lot of information on minimal residual disease sampling and better define this high-risk population of who's at risk for relapsing after CAR T.
Great. Thanks so much, Brian. That was great. In the interest of time, there are 2 comments / questions, and so I'm going to ask you to address those in the chat. So great work. And we're going to move on to the next one, which is S2207, randomized phase 2 study of the addition of cell of origin directed therapy for TAFA-based treatment in non-transplant eligible patients with relapse refractory DLBCL. And Dr. Jennifer Amengual from Columbia University is going to present this.
Okay, thanks so much, Sony. So first, I just want to thank everyone from the group. This is a big team effort and was led by Dan Persky, and who will be leaving us, but we're happy to keep the ship moving along. And so both myself and Patrick Reagan will be really pushing this project along with Hader and Alexi also working on different components. Next slide. Okay, so just as way background, as you all know here, the majority of patients with relapsed refractory diffuse large B-cell lymphoma are not really eligible for autologous stem cell transplant or CAR T, and this is really a huge unmet need. And so the study that we're proposing will focus on older patients or those who are frail, who've been exposed to chemotherapy, and really, can't get further intensive therapy. And so in order to really understand how salvage types of therapies or palliative therapies work in these patients, we'll be incorporating PROs, which we think is really essential to measure quality of life. We know that Tafa-Len is an effective, well-tolerated treatment that's non-chemotherapy based and is FDA approved for non-transplant eligible diffuse large B-cell lymphoma in July 2020. We also wanted to take advantage that diffuse large B-cell lymphoma is classified by different cell of origin. As you guys know, germinal center B-cell and activated B-cell, which is incorporated into the WHO classification. And we want to try to use cell of origin specific agents to improve upon the Tafa-Len backbone in order to improve PFS in these patients. Next slide. Essentially, we proposed a 3-arm study randomizing the control Tafa-Lenalidomide to Tafa-Len plus tazemetostat and Tafa-Len plus zanubrutinib. Patients would be randomized 1:1:1, stratified by cell of origin, GCB versus non-GCB. And in this way, we'll be able to see if the cell of origin directed therapies actually have an enriched response for those cell of origin. Next. And just briefly, the key eligibility will really be pretty much anyone with relapsed refractory diffuse large B-cell lymphoma. They must have cell of origin determination by immunohistochemistry. They must have measurable disease, and they must not be a candidate for autologous stem cell transplant or CAR T. They may have received a prior transplant or CAR T, and patients are allowed to have up to 5 prior treatments. Next. And essentially, the regimens would be simply Tafa-Len as it's given on label, with Tafa given in cycle 1 on days 1, 4, 8, and 15, followed by days 1, 8, and 15, and then on day 1 and 15, and lenalidomide 25 milligrams orally on days 1 through 21. And we'll be giving treatment only for 1 year, so it'll be limited. In arm 1, we'll include a tazemetostat to this arm. We'll have a safety run-in looking at ranges of doses between 600 to 800 milligrams. And this will be given twice daily continuously. Next slide. And in arm 3, we'll be incorporating zanubrutinib as the experimental arm, giving 160 milligrams twice daily. And there will also be a safety run-in for the zanubrutinib tafelen arm as well. And our primary endpoints are to look at progression-free survival and between the controls and each of the experimental groups. And we'll stratify this by cell of origin. And we'll also be looking in secondary endpoints, overall response rate, CR, PR, duration of response. And importantly, we'll be looking at frailty measures and exploratory endpoints such as tumor mutational profiles, ctDNA, to try to understand the effects of these drugs early on. Next slide. Okay, and so with that, I mean, this is very early on. We have submitted this study. We're hoping to hear back from some of the feedback from the CTEP and the steering committee today, and hopefully we'll get some positive news. We look forward to bringing this forward.
I believe they're meeting in 40 minutes. So everybody think good thoughts. And by the end of this meeting, hopefully things will keep moving forward.
Yeah, and I do want to reiterate, just the team effort that's gone into this, and really excited to see this come to fruition. So good luck.
Thanks.
All right. Next, we have Dr. Teisel Phillips from University of Michigan discussing randomized phase 3 trial of continuous versus intermittent maintenance therapy with zanubrutinib as upfront treatment in older patients with mantle cell lymphoma.
Hi, thank you. I'll briefly go over our concept that's actually with protocol being reviewed at this point. So with elderly patients with MCL, there isn't a true definition of the standard of care, but we've always tried to improve treatment outcomes and also safety. So in this situation, sort of pulling from what we've done with CLL, the idea here is to use a completely chemo-free induction regimen in these patients, and also evaluate in certain patients if we can actually stop therapy. So in this, we will use zanubrutinib, rituximab, and venetoclax. So this would be for patients who are 70 years of age or older, or who are 60, between over to 60 and less than 70, who have comorbidities that would preclude utilization of autologous stem cell transplantation. So, I mean, for this, we did pull some of those restrictions from the current zanubrutinib rituximab versus BR study that's currently ongoing. So, as you see from the initial the initial laboratory values as far as ANC platelet count, creatinine clearance, and liver numbers. Next slide, please. So here is the schema, which sort of dictates how we would plan to treat these patients. So these patients would start off initially with zanubrutinib and rituximab. And those who obtain a complete remission, there is a 1-to-1 randomization until continuous zanubrutinib, where they would get that indefinitely, or a retreatment arm, whereas those patients would go into observation, and then they would be reinitiated on zanubrutinib upon clinical progression. For those in a continuous arm, we will follow for a PFS of a year. For those in the retreatment arm, a PFS, you'd be evaluated for an initial PFS in the continuous arm, and then the second PFS for those in a retreatment arm. For those patients who do not obtain an adequate response to the zanubrutinib rituximab, there'll be a second 1-to-1 randomization to those who continue on zanubrutinib or with the addition of venetoclax. Treatment is to continue to disease progression or unacceptable events, except for the retreatment arm, again, where those patients will be followed and observed, and then reinitiate on therapy upon clinical progression. So the rituximab and zanubrutinib combination will be for 6 cycles, similar to the current zanubrutinib BR study.
We have time for a couple of questions. Can I just ask, so
Sure.
the CR is going to be determined then after 6 months, is that right? And that's where the randomization will happen?
That is correct.
What about people who enter a CR later? Are they looking, I'm sure this would obviously complicate it, but it's just the indefinite version. Is there like or do we is there a reason to pick 6 months is, I guess my question, as opposed to like 12? Give
So
people
this,
a little bit longer.
right. So the 6 months was really tried to, as much as we could, we had to pick a time point. And because of the current study, which would be 6 cycles of this combination versus 6 cycles of BR, we tried to match that up as much as we could just to keep some consistency, because that was sort of what was preferred by the steering committee, that we follow that current study of as much as we could, just because while a fair number of us consider the BTK inhibitors and rituximab to be an adequate option, the steering committee was more so focused on actually utilizing a regimen that would be consistent with what's currently being evaluated in phase 3 studies. So for that reason is why we stuck with the 6 cycles.
Great. And we have a question, Dr. Hoffman.
Would there be any consideration to adding P53 mutated patients who otherwise wouldn't meet the inclusion criteria?
So as of right now, they are not excluded.
I'm just thinking if you had someone that doesn't otherwise meet the inclusion criteria, but who has a P53 mutation that historically, based retrospectively, would not likely benefit from chemoimmunotherapy and transplant-based strategies.
So we tried to keep the inclusion criteria as loose as possible to try to capture as many patients. Obviously, we have to draw the line somewhere with some of these values. But the hope is, like I said, we did make it loose and also included some criteria for patients whose maybe lab parameters are impacted by the disease and not necessarily comorbidities per se. But I mean, my experience, I mean, with the other study is that, I mean, the criteria is fairly loose, so we should capture most patients. But I agree, I wish we could put there, I mean, obviously we could just take all comers irrespective, but I mean, we have to have some sort of parameters, unfortunately.
Great. Thanks so much. Okay, so we're going to move on to the last of our new concepts. This is randomized phase 3 interim response adapted trial comparing standard therapy with immuno-oncology therapy for children and adults with newly diagnosed stage 1 and 2 classical Hodgkin lymphoma. We have Dr. Boyu Hu from University of Utah to present this.
Thank you so much, Sony. I have the pleasure of presenting AHOD 2131 to you all this morning. So early stage Hodgkin lymphoma is a highly curable disease with reported cure rates of up to 80 and 95%, depending on the clinical trial. Given there's a large proportion of patients who are children, adolescents, and young adults, we must balance these cure rates with acute and long-term toxicities of therapies. These long-term toxicities have traditionally included infertility, secondary malignancies, and cardiac and pulmonary impairments. Currently, the standard of care includes chemotherapy, mostly with ABVD, plus or minus radiation. While radiation oncologists have avoided the more toxic mantle field radiation in favor of less toxic involved site radiotherapy, long-term toxicities of radiation include secondary malignancies and accelerated cardiac and vascular disease. Therefore, multiple clinical trials have tried to avoid radiation therapy to reduce long-term toxicities and have included using prognostic biomarkers such as PET2 to drive decisions of whether to avoid radiation or not. Interim PET scan after 2 cycles of chemotherapy has been shown to be highly prognostic, in which patients who achieve a complete radiographic remission have better survival outcomes than patients who are PET2 positive. Multiple clinical trials have utilized PET2 to either de-escalate treatment for patients who are PET2 negative or escalate treatments for patients who are PET2 positive. The question is, do we have to escalate to escalated BIACOP? And that's where novel immuno-oncology agents come into play. These novel immuno-oncology agents, such as antibody-drug conjugates, rituximab vedotin, and also immunotherapy, such as nivolumab, have really changed the treatment paradigm of Hodgkin lymphoma. In recent clinical trial, the combination of BV and Nivo has been shown to be both safe and efficacious. And so the hypothesis of AHOD 2131 is, can we incorporate novel immunotherapy agents to not only maintain the high cure rates that we historically have seen with early stage Hodgkin lymphoma, but also reduce the acute and long-term toxicities of chemotherapy and radiation therapy, all the while utilizing PET2 to potentially figure out who would benefit the most from the immuno-oncology agents. So the study design includes early stage Hodgkin lymphoma patients with stage 1 and 2, ages 5 to 60. The favorable and unfavorable prognostics for early stage is according to ERTC, as you can see there. In addition, we'll be also utilizing bulky disease, which is defined as 10 centimeters or greater. There will be anticipated 594 favorable risk patients included in this trial. All patients will receive 2 cycles of ABVD followed by an interim PET. PET2 negative disease is defined as a 5-point score of 1 to 3. All PET2 negative patients will be randomized to either receiving an additional 2 cycles of ABVD or 4 cycles of BV-nivo. I would like to point out that all patients who are PET2 negative will avoid radiation. PET2 positive patients will receive 2 cycles of escalated BIACOP followed by involved site radiotherapy, or BV-nivo for 4 cycles followed by involved site radiotherapy. The stratification factors include age, and then with additional stratification factors for PET2 negative patients of 5 PS score of 1 to 2 versus 3, and a stratification for PET2 positive patients of 4 versus 5. In unfavorable patients only, there will be additional stratification of bulk versus no bulk. Very similarly to the favorable patients, the unfavorable patients will also receive 2 cycles of ABVD followed by interim PET scan. PET2 negative patients will be randomized to ABD for 4 additional cycles and BV-nivo for 4 additional cycles. All of these PET2 negative patients will avoid radiation as well. PET positive patients will receive escalated BIACOP for 2 cycles followed by involved site radiotherapy, or BV-nivo for 4 cycles followed by radiotherapy. So just some of the study takeaways. This is an incredibly large study. It will be a large effort with anticipated accrual of 1,782 patients. The majority of these early stage patients will avoid radiation, as the anticipated PET2 negative number is around 1,500 patients. The primary endpoint is 3-year superiority in progression-free survival, and the secondary endpoint is 12-year non-inferiority in overall survival. And really only a minority of patients will receive escalated BIACOP, which is anticipated to be about 134 patients, or 7.5% of the overall cohort. The study, along with S1826, will harmonize pediatric and adult Hodgkin lymphoma treatments. And as we've seen from S1826, adult treatments can be both safe and efficacious in pediatric patients. And then the majority of patients will hopefully receive reduced chemotherapy strategy in the front line. And this will be one of the few trials that will evaluate that. And the long-term follow-up of 12 years allows us to assess late-term effects of treatment. And we just got comments back from CTEP with an anticipated activation in July of this year with an anticipated 5-year accrual. On behalf of the study chairs and also the champions from our various cooperative groups, thank you, and I can take any questions.
That's great. That's really exciting about the timeline. So, Hildy?
Yeah, this is a really interesting study. What is your strategy to follow up with patients for 12 years? Because this comes up repeatedly. How are we going to find them?
Yeah, that's the hard part. Right now, it's just yearly phone calls to follow up with them for a survival sweep.
Yeah, that is challenging with a young group that's very mobile and
Exactly.
changes names.
Right.
Great.
I'll just mention this, Sandy Counting, that the radiation will be 30 gray, but there'll be an additional boost to the areas that are PET avid at the time of the interim scan.
Thank you. Thanks, Sandy. Thank you, William. That was great. Okay. We are going to move on now. So those were some of our new concepts. And what we've asked for our next speaker, this is Dr. Manali Kamdar from the University of Colorado, where she is she's an associate professor and director of the lymphoma program there. And we've tasked her in 20 minutes to cover what happened at ASH. I mean, as all of us know, ASH was really huge for lymphoma this year with presentations in the plenary and late-breaking abstracts and many other really exciting data that were presented. So with that, I am going to turn it over to Dr. Kamdar to give us an ASH review on newly diagnosed and relapsed diffuse large B-cell lymphoma.
Thank you so much. Hi, everyone. My name is Manali Kamdar, and I'd really like to thank Dr. Friedberg and Smith for giving me this opportunity to talk about some exciting, promising updates in newly diagnosed as well as relapsed refractory diffuse large cell lymphoma from the ASH meeting. So as many of you are aware, RCHOP has been unfortunately, the standard of care for over 20 years, despite many attempts to improve upon R-CHOP as the backbone. On the left is the Goya study, which is the most contemporaneous study, which shows that patients with R-CHOP continue to relapse and that we need to do better for patients who relapse or are upfront refractory to R-CHOP. Polatuzumab vedotin is an anti-CD79B antibody drug conjugate, which in combination with Mustang rituximab has been FDA approved for multiply pretreated relapsed refractory diffuse large cell lymphoma. Given the exciting responses with polatuzumab, next slide, was born the POLARIX study. This was presented by Tilly et al., which basically is a randomized phase 3 study comparing polatuzumab vedotin plus R-CHP versus R-CHOP in patients with previously untreated diffuse large B-cell lymphoma. This is the study design and baseline characteristics tested in previously untreated DLBCL patients between the age groups of 18 and 80 with an IPI of 2 to 5 and an ECOG performance status of 0 to 2, randomized 1:1 to either 6 cycles of Pora R-CHP, followed by 2 additional cycles of rituximab versus 6 cycles of R-CHOP in a similar fashion. Primary endpoint of the study was investigator-assessed progression-free survival, and the key secondary endpoints were event-free survival, complete response rate, and other. The baseline characteristics, going back one slide, were similar between the two groups, between R-CHOP as well as Pola R-CHP. A total of 440 patients were randomized in both arms, and as you can see, with respect to IPI score, field of origin, especially BCL2 expression, and previous rearrangement, the groups were equally spaced out. Next slide. The primary endpoint was progression-free survival, which at a median follow-up of 28.2 months is met with Pola R-CHP showing significantly improved PFS over R-CHOP with a hazard ratio of 0.7 that translated into Pola R-CHP demonstrating a 27% reduction in the relative risk of death, disease progression, relapse, or death. The 2-year PFS with Pola R-CHP is 76.7% versus 70.2% with R-CHOP. Next slide. With respect to other key secondary endpoints, that was also statistically significant in patients receiving Pola R-CHP with respect to overall response rate, complete response rate, investigator-assessed EFS, as well as investigator-assessed disease-free survival. When it came to toxicity with results to, overall survival, at this time, at this median follow-up, there is no difference in the overall survival between Pola R-CHP and R-CHOP with a hazard ratio of 0.9. Next slide. This is the safety summary, and as you can see, the safety profiles were very similar between Pola R-CHP and R-CHOP, especially when it came to grade 5 events, as well as serious adverse events and adverse events that led to discontinuation of any study drug. Next slide. Thus, Pola R-CHP significantly prolonged PFS compared with R-CHOP in patients with intermediate and high-risk previously untreated DLBCL, with safety profiles being very comparable between the two groups. Exploratory analyses currently are ongoing. Pending regulatory approval, the authors did conclude at the end of the presentation that these data support consideration of Pola R-CHP initial treatment option for patients with newly diagnosed DLBCL with an IPI of 2 to 5. Next slide. So shifting gears, let's talk about relapsed refractory diffuse large B-cell lymphoma in the second-line setting. On the left, algorithm that all of us follow in our clinics, which is patients who relapse or are refractory to R-CHOP with newly diagnosed DLBCL, if they remain transplant eligible, we typically give them salvage second-line chemotherapy, either with RICE, RDHAP, or RGDP. If they were to demonstrate chemosensitivity in the form of complete response or partial response, we take them over to an autologous stem cell transplantation. On the other hand, if they were to fail an auto transplant or were not to be chemosensitive to salvage second-line chemotherapy, at this time, the FDA approved CAR T-cell therapy in the third-line setting. On the right is the graph that actually shows the contemporaneous issue with second-line treatment is. This is the CORAL study, which looks at second-line chemotherapy followed by an autologous stem cell transplant in patients who are progressing within 1 year. All of our patients receive our chemo. And as you can see, going back one slide, patients who ended up getting prior rituximab, their overall survival with second-line chemotherapy and possibly autotransplant is extremely dismal. So this constitutes the new unmet need in patients with relapsed refractory DLBCL in the second-line setting. Given the benefits of CAR T-cell therapy in the third-line setting, it was intuitive to test CAR T-cell in the second-line setting. Next slide. And thus were born the concepts with respect to the 3 randomized trials that were presented at ASH 21 that compared CAR T-cell therapy versus standard of care in transplant-eligible diffuse large B-cell lymphoma who have high-risk disease with respect to either being primary refractory or relapse within 12 months after frontline ARC chemo. ZUMA-7 compared axicabtagene to standard of care, Transform compared lisocabtagene to standard of care, and Belinda compared tisagenlecleucel to standard of care, which constituted salvage chemo followed by a transplant. Next slide. So I'm going to quickly go over the study schemas. As you can see, ZUMA-7 enrolled the highest number of patients, about 359, over nearly 75 sites worldwide, and they were randomized to getting either axi-cell or standard of care. Important to highlight that bridging can only be given with steroids on this trial. Crossover was not allowed on this study. Primary endpoint was event-free survival, with the key secondary endpoints being response rate and overall survival. Nearly 75% patients in both arms had primary refractory disease, 16% with double hit and triple hit lymphoma. Next slide. At a median follow-up of 24.9 months, the primary endpoint of event-free survival was met, with axi-cell showing statistically significant superiority over standard of care at a hazard ratio of 0.3. The median EFS being 8 months versus only 2 months in the standard of care arm. The 2-year PFS was 2-year EFS was 40.5% versus 16.3% in the standard of care. Next slide. The median overall survival at this point at the interim analysis was not reached for axi-cell versus it was only 35.1 months for patients who received standard of care. Next slide. This was the schema for the Transform study. It was similar to the other studies. However, key differences were that other patients with Transform lymphoma from non-follicular histologies, primary mediastinal B-cell lymphoma, T-cell rich B-cell lymphoma, secondary CNS lymphoma were allowed on this study. There was no minimum absolute lymphocyte cutoff for enrollment. At the same time, bridging therapy with 1 cycle of chemo was allowed, and crossover was permitted on this study. Similar to the ZUMA-7 study, primary refractory disease constituted 75% and double hit, triple hit 24% on this trial. Next slide. This was a pre-specified interim analysis at a median follow-up of 6.2 months. The event-free survival primary endpoint was statistically significant at 10.1 months versus 2.3 months in the standard of care arm. Next slide. The overall survival curves at this point, they are not statistically significant. However, you can see that the Kaplan-Meier curves are clearly split in favor of liso-cel, and this is despite a crossover. So longer-term data is awaited on this clinical trial. Next slide. And this is the Belinda study. Again, tisagenlecleucel to standard of care. However, there were some key differences with respect to definition of event-free survival, time to infusion, as well as the number of bridging chemotherapy cycles that could be given. Primary refractory disease again constituted 3/4 of the patient population. Double hit, triple hit lymphoma slightly higher in the CAR T arm at 19% versus 11% on the auto transplant arm. Next slide. And very interestingly, no difference was noted on this study with respect to the primary endpoint of event-free survival, where both arms performed fairly dismal at 3 months event-free survival. Next slide. Despite all the issues with cross comparison, this is a key, this is a highlight of all the responses as well as toxicity data with ZUMA-7 and Transform. And to highlight, next slide. As you can see, the median follow-up is the longest with ZUMA-7. With respect to the positive studies, ZUMA-7 or Transform, you can see that the auto-transplant arm is doing fairly poor at a median EFS of 2 to 2.3 months, versus the median EFS is between 8 to 10 months on the ZUMA-7 and Transform studies, respectively. Important to highlight with respect to toxicity, grade 3 or higher CRS and neurotoxicity were at 6% or 21% respectively on ZUMA-7 versus only 1% and 4% on the Transform study. It's important to highlight that the median time from registration to CAR T infusion on the ZUMA-7 study was 29 days on ZUMA-7, 34 on Transform, and a median of 52 days on Belinda. It may or could have influenced the outcomes of this study. Most importantly, prior to me submitting these slides, the FDA approval came forward for Axi-cel, and at this time, Axi-cel is approved for patients who have relapsed within 12 months after frontline R chemo or are primary refractory to R chemo. So this is exciting data that has come out of the 3 studies that were presented at ASH 2021. Next slide. So at this point, the conclusions, of course, were that the 2 studies demonstrated superiority, but Belinda did not. Toxicity was manageable. It was also important to highlight that there were stocks that were analyzing quality of life with respect to CAR T cells for ZUMA-7 as well as Transform, and the accompanying analysis actually showed improved quality of life in CAR T cell arms for both ZUMA-7 as well as Transform. Next slide. So clearly, CAR T cell therapy does well in the 3rd-line setting. Now we know we have an approval for the 2nd-line setting. So then this study, which was presented by Dr. Nilapu and colleagues, looked at the primary analysis of ZUMA-12, which was a phase 2 study of axicabtagene ciloleucel as frontline treatment for patients with high-risk large B-cell lymphoma. Next slide. There's a study design which looked at high-risk large B-cell lymphoma that constituted high-grade B-cell lymphoma with a MYC and BCL2 and or BCL6 rearrangement, or large B-cell lymphoma with an IPI score of 3 or more anytime before enrollment. Plus, these patients had to have a positive interim PET, which would be a Deauville score of 4 or 5 after 2 cycles of anti-CD20 monoclonal antibody and anthracycline-based regimen. They underwent leukapheresis. At this time, they could actually get non-chemotherapy bridging, followed by conditioning. The primary endpoint of this study was investigator-assessed complete response rate. Next slide. These were the baseline characteristics. As you can see, 40% of patients had double or triple hit lymphoma, 78% of patients had an IPI score of 3 or higher, 48% of patients with a score of Deauville 4, and 53% with Deauville 5. Next slide. The overall response rate at this time, with a median follow-up of 15.9 months, was an 89%, complete response rate of 78%. Also important to highlight that there were patients who converted from partial response or stable disease to CR, nearly 16% converted from PR to CR, and 3% from stable disease to CR. This is at a median follow-up of nearly 16 months. Next slide. The duration of response, event-free survival, as well as progression-free survival, Kaplan-Meier curves here, have not been reached at this median. Overall survival median is 24.5 months. Next slide. With regards to safety, the most common axi-cell-related grade 3 or higher adverse events were neutropenia, seen in nearly 50% of patients, anemia in 30%, 15% encephalopathy. CRS rate, any grade, was 100%, with grade 3 or higher being more than 8%, and no grade 4 or grade 5 CRS events occurred on this clinical trial. Neurological events, any grade, was 73%, 5% had grade 4, no grade 5 neurological events have so far been reported. Next. With respect to CAR T cell expansion, it certainly appeared to be much greater in ZUMA-12 compared with ZUMA-1, thus basically probably suggesting improved T cell fitness as we move earlier along the CAR T cell paradigm. Next slide. So this is again a cross comparison across the third line CAR T setting in ZUMA 1, second line in ZUMA 7, and front line in ZUMA 12. Although too early, given the median follow-up is short on ZUMA 12, I cannot help but compare the overall response rates and complete response rates may look very similar. The median progression-free survival seems like it may look better as we keep getting CARs earlier in the course of therapy, with a 12-month PFS of nearly 75% on ZUMA 12, 55% on ZUMA 7, 44% on ZUMA 1. So with that, I'm going to go to the next slide. So clearly CARs have made quite an impact, but as all of us know, about 60% of patients will not really get the long-term benefit of CAR T-cell therapy with the existing CD19 products. The post-CAR T relapse primarily constitutes 70% of early onset with CD19 positive disease, whereas late onset 30% with CD19 negative disease. This is intuitive, but it's important to see that now there is data which shows that patients who relapse after CAR-19 therapy, their responses are really poor, with a median PFS of only 1.8 months and a median overall survival of only 6 months. So this constitutes the new unmet need in our field. Next slide. There were several abstracts that were presented at ASH 2021, looking at many, many different ways to improve upon existing CARs, to think about allogeneic CARs, bicistronic, bispecific CARs, as well as bispecific antibodies. I had to pick only 2, so this is what I picked. And this is a study that was presented by the group at Stanford, led by Matt Frank, with a CD22 CAR T-cell therapy in adults with large B-cell lymphoma who have relapsed after CD19 CAR T-cell therapy. This was a phase 1 study with the primary endpoint being feasibility, finding the phase 2 dose and safety and toxicity. The eligibility criteria included relapse refractory large B-cell lymphoma who have relapsed after prior CAR-19 therapy or have CD19 negative disease. Next slide. This is that is important to highlight. With CAR-20, the CAR-T associated toxicity was extremely manageable. With respect to CRS, grade 3 toxicity only occurred in 1 out of 24 patients, grade 2 neurological events in 2 out of 24. It's important to highlight that there is an emerging CAR-22 related toxicity, the macrophage activating syndrome, or the HLH-like toxicity, which was actually seen in 5 out of 24 patients. This is something that we begin to learn as investigators as we test these novel constructs. Outside of that, there was only 1 grade 5 event from sepsis that led to multi-organ failure. Next slide. With respect to responses, the clinical outcomes in this study were very promising or looked promising. Overall response rate of 79%, overall complete response rate of 58% at a median follow-up for the whole cohort of 8.6 months. Overall, the median progression-free survival as well as overall survival has not been reached, albeit the median follow-up is under 12 months. This certainly looks like something that we will have to watch out for. Next slide. Switching gears to the last topic of this talk would be the bispecific antibody treatments in patients with relapsed refractory diffuse large cell lymphoma. Dr. Schuster had previously presented mosunetuzumab at an ASH plenary, which led to unleashing of the construct, especially in patients who had progressed after CAR T-cell therapy. Next slide. This is a busy slide, but I had to pick and choose amongst all these bispecifics that were presented. And this is the key takeaway of these 5 clinical trials that were presented using glofitamab, subcutaneous mosunetuzumab, mosun-pola, glofitamab-pola, and then there's an IgM-based bispecific antibody called IgM2323 that was all tested in patients with relapsed refractory large B-cell lymphoma, as well as some other subtypes. Important to highlight that the median line of therapy in most of these clinical trials were 3. Median follow-up at that point is the longest in glofitamab at 13.4. Mosunetuzumab plus polatuzumab was something that I would have to say had some exciting results, especially in patients who had failed post-CAR T. So with prior CAR T cell therapy, you can see that in GLOFI, the overall response rate for the whole cohort was 54%, complete response rate of 39%. The CRS all grade was about 59%, but grade 3 or higher was only 5%. ICANS 2% with absolutely no evidence of grade 3 or higher ICANS. On the other hand, if you look at the mosunetuzumab plus polatuzumab study with a total of 63 patients, you can see that the overall response rate was 65%. In the post-CAR T cell therapy setting, which were 24 patients, 62% had response. The CR rate overall for the cohort was 48.3%, and 10 out of 24 of the post-CAR T patients had a complete response. The median duration of response was not reached. Complete cytokine release syndrome was seen in 18% all grade, no evidence of grade 3 or higher CRS. On the other hand, with ICANS, all grades were seen in 8%, with 3% grade 3 or higher. So overall, I think this looks very promising with respect to all of these studies. Longer follow-up is obviously awaited, but it definitely shows some promise, especially in patients in the post-CAR T space. Next slide. So overall, I do believe that bispecific antibodies are here to stay because they did show high response rates as monotherapy as well as in combination in patients with relapsed refractory, heavily pretreated patients, including post-CAR T cell therapy failures. Adverse events at this time look very manageable. These are off-the-shelf products, so the logistical challenges around manufacturing, etc., technically get completely mitigated with bispecifics if you were to compare it to CAR T cell. Obviously, long-term follow-up is required. But with this, I do come to the end of this presentation. Huge advances are coming. We're just so delighted to be investigators in this day and age to advance the field in patients with relapsed refractory large B-cell lymphoma. Thank you very much.
Manali, that was great. So much exciting work being done in lymphoma. This is open for questions. I think we have time for one or two. I have just sort of maybe a detailed question, but in Zuma 12, as we, think about moving CAR T up earlier and earlier and earlier, I can't help but think that, of course they're going to do better, right? These are previously untreated patients. Do you know if they're going to look further between Deauville 4 versus 5? Because in some ways, Deauville 4 could be, just slower responders, whereas 5 is overt progression.
So I did ask the same question to Dr. Milapu, and he did say that the Deauville 4s were primarily progressions and they were not partial responses, but let's await the manuscript is exactly what he said.
Yeah. Okay, well, thank you again. That was really great.
Thank you.
All right, we are going to move on. Do you want to take over?
No, I'm Alex.
Oh, you're Alex. I'm sorry.
Can't you tell?
Okay.
Alex has aged and lost a lot of hair.
Well, we're going to move on to the last part of our morning session, which are our ongoing studies. And we have a number of different presentations. They're going to be short, but really give you a highlight and an update as to where we are. So for S1826, I will have Dr. Friedberg covering for Dr. Herrera.
So this is our biggest accruing trial right now. It's a randomized trial in advanced stage Hodgkin lymphoma, open for patients ages 12 and above. This is a harmonized trial between the pediatric group and our group. And it goes across the US intergroup, as well as the Canadian clinical trials group as well. Just to remind people, I think many of you know that this is a 1-to-1 randomization between AVD and brentuximab, or the Echelon 1 regimen, compared to AVD and nivolumab, 6 cycles of each treatment. Radiation therapy is an option mainly for pediatric patients, but if there's an occasional patient that you'd like to give radiation therapy, you can choose to do that at the beginning. And there are strict criteria that need to be followed. And thus far, using those strict criteria, less than 5 patients have actually gotten radiation therapy on this trial. So it's essentially a chemotherapy alone trial. In addition, we're looking at quality of life in a variety of ways, including patient-reported outcomes, not just during treatment, but after treatment. This trial has accrued very well. There are over 800 patients enrolled out of a planned 987 patients. So we expect the trial will, if it keeps accruing at the same rate, complete accrual by sometime in the early fall. This is the current study status. The point to make is that this has been a real Herculean effort across a number of individuals. I will point out just a couple of things. First of all, it was announced in a press release, and I believe will be presented at the ASCO meeting, that in the Echelon trial, the AVD brentuximab arm is now showing overall survival benefit or superiority over ABVD. So I think patients who are randomized to the AVD brentuximab arm on this trial are getting certainly cutting edge therapy and potentially therapy that has been shown to be superior to ABVD as far as overall survival. The second issue that has come up is around toxicities. There are certainly toxicities that are expected on both of these arms, including immune-related toxicities on the nivolumab arm. I can't tell whether they're occurring more frequently than you'd expect because this is a younger patient population, potentially with a more intact immune system. There does not appear to be a signal in the children of increased toxicity with the nivolumab, although that's more anecdotal than a rigorous review. In the book that came with this meeting, you can see a breakdown of all the toxicities. And the question that we keep getting asked for some reason is, can we give these patients Evusheld or other protection against COVID? And the answer is absolutely yes. That's not banned. I'll take any questions on the trial. Great work. This is a heroic, I think, effort among all of the cooperative groups. It's going to end. I have no doubt in my mind that this will define the standard of care for Hodgkin lymphoma in this country and in most of the world moving forward. So a very, very important study.
Thank you. I love the pie chart here, just showing how many different groups are involved. I mean, that's really beautiful. Okay, going to move on to Dr. Elizabeth Brem, who will present S1918. Phase 2-3 randomized trial. Yep, go ahead.
Tough act to follow there, but very happy to give the updates on S1918. So in case anyone is new, this is our randomized phase 2-3 study of our mini-CHOP with or without oral azacitidine in our older patients. Next slide, please. So just the schema is relatively straightforward, other than the fact that the oral azacitidine is kind of given as a priming if you are randomized to that arm. Since the last time we presented this study, we have had a revision go through. So this study has always had a pre-phase. There's evidence, particularly in our older patients, that giving a pre-phase of some kind does improve outcomes. In the first version of this protocol, it was a combination of vincristine and prednisone that turned out to be problematic for a number of reasons. So at this point, the pre-phase is simply prednisone. It is 4 to 7 days. It is 60 to 100 milligrams at the discretion of the treating investigator. The other thing that is a little unique about the study is we are currently in what is called the safety run-in. So the first 40 patients we are watching very closely. We are having calls every 2 weeks, which many of you have been on, which have been incredibly helpful, the feedback we've gotten during those calls. And then after we accrue these first 40 patients, there will be a brief pause to make sure we don't have any huge safety signals that are imbalanced between the two arms. Next slide, please. So again, the key updates here, revision 1 is now active. The prednisone pre-phase is really, I think, the key thing to know. It turns out we did not explicitly address CNS prophylaxis in the first version of the protocol. So now IT alone is allowed, no high-dose methotrexate. And of course, you always miss something. So we're already compiling a list of what needs to be fixed in revision number 2. As I mentioned, we are currently in the safety run-in phase. As of a couple of days ago, we have accrued 26 out of our 40 patients. So the safety run-in at this rate will probably be a few more months. And then again, yes, there will be a brief pause. Anecdotally, I can say that while there have been a couple of strange little things that come up, there do not seem to be any major imbalanced, for example, grade 4 or grade 5 toxicities in the oral azacitidine arm that's going to cause any major issue. But of course, this is why we're doing the safety run-in and why there will be a brief pause, but honestly, so far, so good. So I guess I'll leave it there. And if there's a moment, I can take any questions that come up.
Great, thank you. Any questions at all? Yep, this is exciting. We're in the home stretch for the safety run-in and hopefully can go on from there. So thank you.
Really want to push people to start getting this one open. I'm just looking at the list and, you know, as expected, the accrual is starting to ramp up, but I can really only see 3 SWOG institutions that have accrued patients to this. So clearly an unmet need. There are not a lot of competing studies in this area. And as I think, as you heard, this seems to be a very interesting regimen with a lot of rationale. So this is one to really try to go back and get open so we can get through the safety run-in and really start the randomized question.
Great. We'll move on to Dr.
Just
Barr. Oh, sorry.
Just a quick reminder, it's all comers age 75 and older. So age is the only cutoff, no cell of origin, nothing. Anybody age 75 or older is potentially eligible.
Yeah, including the double hit, right?
Yes, absolutely.
Okay.
Good morning. So wanted to give you a quick update on S1608. Thank you, Sonny. This is the randomized phase 2 trial in early progressing follicular lymphoma. There we go. So just a quick reminder, these are patients with follicular lymphoma who progress within 2 years of first-line chemo-based therapy, including bendamustine or CHOP, randomizing these high-risk patients to umbralisib-based, lenalidomide-based, or additional chemotherapy-based treatment combined with obinutuzumab. And we're really focusing on CR rates after 6 cycles of therapy and have a number of translational endpoints built in as well. And I think many of you have seen this many times previously. We tried to amend the inclusion criteria over the years to be as inclusive as possible. Well, I'm not sure these are the most recent slides, so I'll just give you the update. The big update right now is that the study is on pause. Globally, all studies with umbralisib were put on clinical hold by the FDA. And the reason for this is that there's been some safety concerns in CLL clinical trials, not only with umbralisib, but with other PI3K inhibitors. So as of right now, we can't enroll to this study, but later this month, there will be ODAC meetings focused on the PI3K inhibitor class on the 21st of April, followed by an umbralisib specific meeting with the FDA on the 22nd. So we'll learn more very shortly about how this trial should proceed. But because of the original design, the study will be able to continue as previously planned with all 3 arms. If the FDA feels that this and other clinical trials shouldn't go forward, it still would be possible to continue and finish up this study with the 2 arms only, lenalidomide and the chemo-based arms. So more to come, but we still do have a path forward for this study. About a little more than 2/3 of the study have been accrued, so we're hoping that we can finish this up after we get this reactivated. Any questions?
Just to emphasize, in the book, as you can see what's been published, we have not seen a safety signal across the 3 arms in the patients that have been enrolled.
So we'll move on to the next.
What's the current enrollment?
Are there more questions for Paul?
The question was, what is the current enrollment?
The current enrollment is 72.
Thank you.
So, okay. Dr. Stiff.
Good morning. I'm here to update the Alliance-led A051310. Oh, great. Thank you. I think I still had the training wheels on my bike when this opened. This actually was opened in 2016. Everybody knows the design. This is an auto transplant plus or minus ibrutinib for activated B cell subtype. When this trial initially opened, we were using nanostring precision medicine diagnosis for the ABC subtype. We ran through the funds available and enrolled about 30 patients. A lot of frustrations, many of you know. Trial was closed for a year and then reopened in 2019, I believe. We're now using the Hans algorithm. The trial is accruing very slowly, and obviously with ZUMA-7, there is some concern about this trial ever being completed. This was originally a registrational trial. And again, just to show you that the schema here is if you have relapse refractory DLBCL, you could be within 1 year of salvage of relapse or initial therapy. You have to have salvage chemotherapy and have a PR, which was always the criteria for moving forward with auto transplant. And if the case, you were randomized between transplant plus ibrutinib during the preparative regimen, and then it was discontinued until engraftment and then continued for 12 months, or a placebo. So the primary endpoint, PFS at 2 years. This was, again, originally a 296 patient clinical trial, two-sided alpha 0.05, power 82%. Obviously, this trial was not going to get done. It was no longer going to be a registrational trial. And so the two options were to redo the statistics to see if we could get something out of this, or to close it completely. Alliance went to CTEP and they worked out a new arrangement. So this is going to be a one-sided alpha 0.1 with a power of 74%, and the number dropped from approximately 300 to 160. The good thing is that lowering the numbers allowed for the planned interim analysis for this trial. So an interim analysis then was initially, I think, going to be 80 to 90 randomized patients with 2 years follow-up. We dropped it in half, so now 40 patients. That analysis is ongoing. I checked just before the meeting again. The results are not in. So the DSMB may say, hey, there may be something here, let's keep going. And if that's the case, we want to continue accruals. We're getting about 2.5 patients per month, and with that, 30 months to accrue. So 108 have been pre-registered since the revision in August of 19. 57 patients have been transplanted. So we know those patients are out there. We don't think that this is going to interfere with what's going on with ZUMA-7. I don't think anybody would transplant somebody, and they're not eligible for this if they don't have a PR to salvage therapy. And I think most of us, I think when we see a relapsed patient, they're going to give them some sort of chemotherapy, even if we're thinking about giving them CAR T cells. But if they have a fantastic response, who knows, this may actually be better than a CAR T therapy, certainly a lot less expensive and potentially a lot more safe for patients to receive this without a lot of toxicity, morbidity, hospitalizations. And so I would encourage everybody to continue to try to get patients onto this trial. Other maintenance trials, we know the BV Hodgkin's trial improved progression-free survival by 20%, and who knows, giving ibrutinib to this patient population might actually be superior to giving them something like a CAR T product. So again, encourage you to try to participate, and I'm happy to answer any questions regarding this. Getting the patients on the trials very quickly. The slides are sent to Eric C. at Cleveland Clinic. You get turnaround within 3 to 5 days. You can even send unstained slides or stained slides. If you have the antibodies already done, your pathologist can send the stained slides. Eric will take a quick look at them and say, yes, this patient is eligible.
Just, you brought up the cost. I think that's going to be really helpful, looking at the cost and the safety. Do you think the interim analysis will be presented at ASH then, or submitted to ASH?
I'm sure. Whether the question is whether it's, I don't know the dates for the late breaking. Probably it's passed for ASCO, but I do think they are going to let us know, and they're going to say the trial will continue, or we're going to stop the trial at that point in time. And obviously, I think they're going to do that with CTUP's input.
Great. Thanks so much. Thank you. We'll go on to the next trial update. Randomized phase 2 study of duvelisib or 5-azacitidine with CHOP or CHOEP.
Regarding safe dosage.
Sorry. Okay, that might have been an error. Are there questions online? Okay. All right, I'm going to turn it over to Dr. Amangwa.
Okay, thanks so much. I'm really excited to present this study. You may have seen this before, but this has newly been activated and just starting to get rolling. Next slide. Okay, so as you all know, PTCL is extremely rare and heterogeneous, and patients overall do very poorly with a 5-year progression-free survival of only about 20 to 30% for most subtypes. Median survival after first relapse is pretty dismal, with a median survival of 6 to 10 months only. Next slide. So based on this, a group of us have been discussing how we could potentially improve outcomes in patients with PTCL. There have been studies evaluating CC486, which is an oral azacitidine and approved for MDS and AML, in combination with CHOP, just been performed by Gia Ruwan. And they found that there was an overall response rate of about 85% and 55% CR rate. And this was enhanced in patients with T follicular helper subtype T-cell lymphoma. In addition, duvelisib, which is an oral gamma delta PI3 kinase inhibitor, was studied as a single agent in the PRIMO study with an overall response rate of 52% and CR rate of 36% in relapse refractory PTCL. And we'll see how this plays out based on what Dr. Barr had recently said, that so far this study is continuing on. So with this in mind, next slide.
Sorry.
It's okay.
It's not advancing. There we go. Sorry.
We developed a trial that's looks at these targeted therapies in combination with CHOP or CHOEP for untreated PTCL who are CD30 negative. Recognizing that the Echelon-2 study improved overall survival in patients with CD30 positive PTCL. We wanted to try to improve survival in those who are CD30 negative. The study will stratify for T follicular helper type PTCL and whether patients get a CHOP or CHOEP background, and that will basically be decided based on the patient's age, where those greater than 60 get CHOP and those less than 60 get CHOEP. So patients will be treated with duvelisib plus CHOP or CHOEP, oral azacitidine plus CHOP or CHOEP, or CHOP or CHOEP alone as the control backbone. Next slide. Key eligibility, just to keep this brief, is really newly diagnosed PTCL with less than 10% staining of CD30 by immunohistochemistry. They must have measurable disease and no CNS involvement. And those are really the main eligibility. Next slide. And so for the duvelisib CHOP arm, patients will get duvelisib on days 1 through 21, orally 25 milligrams twice daily on days 1 through 21. Right now, there's a safety lead-in where the first 6 patients will receive duvelisib 15 milligrams BID with CHOP or CHOEP, and 6 with CHOEP. And we'll evaluate toxicity over the first 2 cycles. Go ahead, you can go to the next slide. For the oral azacitidine arm, patients will actually be pre-treated or have like a treatment similar to the elderly study in which patients receive oral azacitidine prior to CHOP on days -6 to day 1, and then subsequent cycles from day 8 to the end of the cycle. So sort of kind of priming each cycle with azacitidine. And again, for the first 12 patients, there'll be evaluation for toxicity. Next slide. And finally, patients on the control arm will just receive CHOP or CHOEP. I indicated here that these orange arrows indicate when ctDNA will be collected. And just to go through the imaging time points, patients will get a baseline, post-cycle 4, and then at the end of treatment. And we'll be looking at PET scan relation to response. And so we're really excited. This has just started. There's only 5 patients on so far in the duvelisib toxicity run-in part of the study. And we look forward to enrolling patients on this. If anyone has any questions, please feel free to reach out to me or Dr. Mehta Shah. Thank you.
Jennifer, do you know if the hold that's affecting umbralisib might be affecting this trial? Have you heard anything about that?
I have not heard that to be the case at this time.
I think it's good to hear your voice. It means you weren't called to the CTEP call, right?
Not yet. I'll let you know.
Another 40 minutes. Okay.
Yep.
Great. Thank you. This is really exciting. So I'm going to go back to Dr. Kamdar to talk about our randomized phase 3 intergroup study in primary mediastinal B-cell lymphoma.
Thank you so much. So as everyone's aware, primary mediastinal B-cell lymphoma is a rare subtype of non-Hodgkin lymphoma, primarily affecting AYA population. The current outcomes with chemoimmunotherapy consists of either R-CHOP plus radiotherapy, which has been historically recommended. However, there's always interest to avoid radiation treatment in a young age group of patients that are afflicted with this subtype of lymphoma. Dose-adjusted EPOCH-R without radiation treatment did show in a single center setting, which was a phase 2 study, that the 5-year EFS was 93%. However, many other multi-center studies that were further done using the same regimen showed that this EFS was not really replicated over 90%, was somewhere between 60 and 80%. Genetic alterations in the primary mediastinal B-cell lymphoma, such as the 9p2.24.1 amplification, has shown that PD-1 inhibition with nivolumab or pembrolizumab has shown promising efficacy as well as safety in patients with relapsed refractory PMB. And thus, this is the design of the ANHL 1931 study, which is a study that is going to be done, is being done in patients with newly diagnosed primary mediastinal B-cell lymphoma. Patients can either receive R-CHOP or dose-adjusted EPOCH-R or R-CHOP plus radiotherapy. However, the physician needs to declare that at study onset. 1 cycle of chemotherapy that may be given in the outside hospital setting is permitted on this trial prior to enrollment. So all of these patients with newly diagnosed PMBCL after registration undergo a randomization in a 1:1 fashion to either getting 3 cycles of chemoimmunotherapy or 3 cycles of chemoimmunotherapy plus nivolumab. Interim PET scan is done after 2 cycles, and as long as they have a response, which would equate to either a complete response, partial response, or stable disease based on Lugano 2014 criteria, they additionally get either 3 more cycles of chemoimmunotherapy alone based on the arm they are randomized to, or 3 cycles of chemoimmunotherapy plus PD-1 inhibition. After 6 cycles of therapy, they undergo an EOT PET-CT. If they have progression of disease, they get off treatment. If they have a complete response rate, if the physician declares it to be a chemo alone arm, then they undergo observation. On the other hand, if they have registered for a radiotherapy arm, that they do end up getting radiotherapy, even if they have achieved a complete response. For patients who have a partial response based on Dovell 4 and 5, if they are chemo alone, they can either undergo observation for a brief while, or they can undergo a biopsy, and if the biopsy is positive, then can undergo radiotherapy. On the other hand, for stable disease, if they are chemo alone, they go off treatment. For R-CHOP plus radiotherapy, they undergo radiation treatment. Next slide. Next slide. So overall, the primary endpoint of this study is progression-free survival as determined by the IRC. Secondary endpoint is EFS, overall survival, rate of PET positivity as in Dovell score of 4-5 at the end of treatment PET. Exploratory objectives are going to be done in the Bollard lab. In terms of accrual, the study needs a total of 186 patients. Expected rate is about 50 per year. So far, the study has been open for about 5 months, and only 15 patients have been enrolled, primarily by pediatric sites. Big kudos to Dr. Brem. She has enrolled a few patients on this study already, and the estimated time to accrual at this point is 3.8 years. But given the rarity of this subtype, we really do need a wholehearted effort from all the intergroups in order to make the study a success.
Thank you. Any questions for Dr. Kamdar? Yeah, hopefully now, I'm not sure. I know the pandemic has probably hit some accrual for some trials, but hopefully this will also start to pick up. Okay, we're going to have Dr. Till come back and speak to us about mantle cell lymphoma.
Thank you. Just a quick update on EAA 4151. This is the transplant versus no transplant in first remission for mantle cell lymphoma. This is the schema here, just to remind you. Patients can get enrolled at any point. There's a step 0 where they get enrolled initially to get the diagnostic tissue that you send to adaptive to identify the clonal marker. They then finish their induction, get restaged with a PET scan and a bone marrow. If they're in a CR and their MRD, and then you also send a peripheral blood test to check for MRD. If that's negative and they're in a CR, that's the randomized population. That's about 80% of the patients. And they get randomized to an auto transplant or no auto transplant, and then everybody gets 3 years of rituximab maintenance every 2 months. Everybody else who doesn't have an MRD CR, so if you have an MRD positive or you're in a partial remission, they stay on the study and they get an auto transplant or rituximab maintenance on a different arm. So we're accruing fairly steadily. It's moving along. There's 476 patients who have assigned to a treatment arm. I think there's more like 650 or something who have actually gotten to the step 0 point. And that's to target a total of 689 to get 434 randomized patients. However, there's been a little snag in that fair number of the patients, I think it's about 10 or 15% in the randomized to the transplant arm, have refused their assigned treatment. So there's discussions ongoing that we're waiting to hear from CTEP about whether we're going to increase the total accrual to try to make up for that. So it kind of highlights when you're having this discussion with the patients. I know it's a little bit of a big drastic decision, change to either get a transplant or no transplant, but they really need to be willing to do that. And so I kind of try to pin them down a little bit ahead of time and say, look, if you don't want to, if you have a very strong secret desire to not go on transplant, just don't go to transplant, just don't be on the study. You get to do what you want if you don't go on the study. So, so that's the main point. There's a few other reminders that we allowing patients to skip up to 4 doses of rituximab if needed, or if in the judgment of the treating physician for COVID-19 reasons. My editorial comment is that, given the data that patients within a year of the last dose of rituximab don't respond and don't get an antibody response to vaccines, 4 doses is probably not enough to make a difference there, especially now that we have Evusheld. I would probably just recommend, prioritizing that and staying on track, but you can skip if you want. You do have to make up the doses at the end. We need a certain number of genomes in the denominator to count as a true, to make it truly MRD negative. So just let the white count recover after the last cycle of chemo before you send the MRD test. You can send the MRD test and the tissue. If somebody comes to you after
onto the radar after they've already finished induction and you're trying to move things along, you can send the tissue from diagnosis and their blood at the same time, and Adaptive can sort it out and move them along. So that's, and we are going to, the last thing is, if you do have somebody who refuses their assigned treatment, they don't come off study, they stay on study. We want to keep tracking their survival data, and then we're just going to do a later intent to treat versus as treated analysis. So any questions?
Have the refusals been fairly balanced between refusing transplant versus refusing no transplant?
No, as you might guess. I mean, there have been some people who were assigned to no transplant who secretly wanted the transplant, and then, or for whatever reason, refused that treatment. But most of it's more imbalanced that people got assigned to transplant. They had been kind of hoping for the no transplant and then backed out. So I think it's like 10 or 15%. So
I have one of those.
Yeah.
I tried. Okay. Thank
And I
you.
think sometimes they kind of want to know the MRD test result. And so you can kind of let them know that that can be done off study too. So that's not only reason to go on study.
Thank you. Any other questions? Okay, we'll go on to Dr. Amin Gwalagan again. Yeah, I don't know if there's a whole lot here, but do you want to just give us a one-line update?
Absolutely. So this one's going to be real quick. So basically, this is the study in relapse refractory Hodgkin lymphoma, looking at Brent-Nivo plus Ipi versus Brent-Nivo. This study is now closed to adult enrollment, but has remained open in COG to children. There's been no safety updates that have been provided, and I don't have any, unfortunately, any information about early signals of response, but hopefully we'll hear some perhaps at ASH. So if you have any younger adults that meet the COG criteria, the AYA, I encourage you to enroll young patients, and to encourage your colleagues also to enroll patients to the study.
I think you just, yeah, go ahead.
And so we've done quite well. We've put 20 patients on and SWOG. All right, thank you.
Thanks so much. Okay, we're going to go on to Dr. Spurgeon, and maybe next time we'll put this one before Dr. Till goes.
Is Steve presenting, or is Anusha presenting?
We have, yep, actually, okay, we're going to call Dr. Balarupali to do the next.
I'm here. I realized I'm on mute. Can you guys hear me?
Yes, we can hear you.
Okay, sorry about that. So this is a study that really builds on what Brian was talking about, but it's in reverse order. We're really working to try an induction regimen that's the best for our patients before they go on to transplant in particular. So this is our randomized 3-arm phase 2 study comparing BR-Ara-C, BR-Ara-C plus Acala, and BR-Acala in patients less than 7 years old with untreated mantle cell lymphoma. As we know, bendamustine Rituxan is becoming a widely used treatment standard in promising chemotherapy backbone. It builds on previous work, studies that we've been involved in. And the incorporation of cytarabine into this regimen leads to high response rates, including MRD negative CRs, which allows for successful stem cell mobilization. As an aside, I do want to point out that some patients actually have not mobilized after this regimen, so we have special interest in keeping track of that. It's not many patients, but please keep us posted. MRD negativity after treatment is associated with prolonged PFS, improved overall survival, whether that be using flow cytometry platforms, a number of PCR-based platforms, or next-gen sequencing. And we know that BTK inhibitor is now being evaluated and soon to be reported in the upfront treatment setting with BR and non-chemotherapy-based regimens. The likelihood is that that could be practice changing in combination with BR for patients over the age of 65. So the goal of 4181 is to identify the most active induction regimen in MCL patients aged less than 70. And the secondary goal is to enroll these patients over to 4151. As an aside, patients definitely need to be gently put this way, because the idea of enrolling on a 3-arm study, one of which is outpatient, the others are involved inpatient treatment, and then wrapping their head around the idea of the 4151 transplant study all at once is very difficult. So just a reminder to introduce that later in the course, but the goal is to enroll them into 4151 as Brian alluded to. Next slide. So this is really a phase 2 screening selection trial. So really it's these 3 arms with ARSC, BR being the control arm, trying to look at MRD CR rates as well as toxicity. Ultimately, if the efficacy or MRD CR rates are similar, then toxicity can be used to pick the best arm. But here you can see arm A, which is the control arm. You give 3 cycles of BR followed by 3 cycles of Rituxan ARSC. And ARSC can be administered outpatient, and a reminder, Rituxan can be administered outpatient as well. Many dose reductions for creatinine clearance and based on blood counts. Arm B is essentially the same idea, but it incorporates acalabrutinib. Now remember, so acalabrutinib is given pretty standard how we give it in combination with BR on days 1 through 28. But with cycles 4 through 6, remember, it's given the thrombocytopenia, especially it's given on days 1 through 7, and then days 22 to 28. And importantly, Arm C incorporates a non-ARSC based regimen, which is all outpatient. This is one of the challenges when we do talk to these patients about it, because many of them are really gunning for Arm C. But just a reminder, as Brian alluded to as well, trying to get these patients to really commit upfront to all 3 arms. Rituxan biosimilars are permitted. As I mentioned, ARSC outpatient is permitted. And I think the key thing here is we are quite flexible and aggressive on getting patients on studies. So don't ever hesitate to reach out. The team's been very good about reaching out to us with questions. And sometimes the protocol can omit some details, and that's by design. So it provides some flexibility, but please reach out with any questions. So frankly, this trial kind of started with a little bit of a thud. It got activated centrally, but took a while to get sites open in 2019. And then we had the pandemic, but it has picked up steam again, and it's been enrolling quite well. Target is 369. We've enrolled 247, so almost 70%, with the goal of closing in a year, or closing to accrual. And SWOG has accrued about a quarter of these patients. There are a couple centers in particular that have been enrolling very well, including since the last SWOG meeting in the fall. So Rochester, the Kansas hospitals, and Utah. And then Chicago has continued to enroll. But as we know, these mantle cells fits and starts, but please think of this study. This includes all comers, whether they're high risk or low risk. And I think as much as we're tempted to enroll patients, especially over the age of 60, on sort of novel chemo-free regimens, if they do have high risk karyotype or mutation analysis, I would encourage that question still remains unanswered with these regimens in a prospective setting. So I would not let that have people shy away from enrollment. So just to plug, I think we can get done in time, but I think we have some more work to do. Thank you.
Great. Thanks so much, Steve. Any questions for Dr. Spurgeon? Okay. Yeah, it's nice to see the accrual, and hopefully it'll get completed soon. So, okay. So we're going to move on to AO51701 and invite Dr. Valarapalli to talk about double hit and double expressing lymphomas.
So just a major couple of updates on this study. The study is currently on enrollment hold since August last year. But when it started as a study for upfront untreated double expressor and double hit lymphomas, randomized to either R-chemo versus R-chemo plus venetoclax, with double expressor arm receiving our chemo as backbone, and then double hit would be our DAA-POC for backbone. Pretty standard inclusion and exclusion criteria. With the double expressor arm, it has finished its enrollment, and that's one of the reasons that the enrollment is on hold. When the initial study was proposed, the null hypothesis for the double expressor arm was 24-month PFS of 45% at baseline, and the study was expected to improve the PFS to 65%. But since the study had been developed, we've seen some updated study results, especially with Goya and LLBC data, that the 5-year PFS is much better than what we had thought initially, and it's now around 62%. So there is an independent statistical review going on for this specific subtype. And once the statistics has been updated, it will open to re-enrollment sometime later this year. This isn't still in process. The double hit arm with the DA-EPOCH versus DA-EPOCH plus venetoclax had to close because of the safety signal. This data was presented at ASH 2021 with 5 deaths from septic shock, mainly secondary to neutropenia. So that arm as it is not going to reopen. There has been a lot of discussions going on with NCI and CTAP, and there is a first draft of amendment in process right now, looking at DA-R-EPOCH versus R-CHOP plus venetoclax with a new statistical analysis and design. So be on the lookout for it, hopefully sometime in the next few months for this one. As we know, DA-EPOCH for double hit is only based on 1 retrospective and 1 prospective studies, and there's not a whole lot of randomized prospective studies in this space. So be on the lookout for this one. Thank you.
Great, thank you. Any questions? Okay. Well,
I really hope that double hit gets opened again, because I think it surprised everybody, the rate of accrual. I think we all recognize that's an area that we need to do better. And that was another study that was over accruing to the point where they were initially thinking of maybe a amendment to refine the number of patients going on, make it larger to get a better estimate. But obviously the toxicity has to be ironed out.
it would be great to not have to give an inpatient regimen or an intense regimen if we could use R-CHOP. So, okay, so Dr. Stevens is going to close us out for the day or for the morning, talking about SWOG CLL S1925.
Thanks for allowing me to present this study. It's actually run out of the leukemia group, but as you all know, most of us are likely the people seeing these patients. And so really pertinent for this group because I really need some help with accrual to this study. So this study is a randomized phase 3 study of early intervention with venetoclax and obinutuzumab versus delayed therapy with venetoclax and obinutuzumab in newly diagnosed asymptomatic high-risk patients with chronic lymphocytic leukemia. As I've presented this study a few times, this is really just a reminder. This is for newly diagnosed patients with CLL. Newly diagnosed means that it has been within 12 months of their initial diagnosis. And so you really have to identify these patients early in their course. These patients must be asymptomatic, not requiring therapy by IWCLL criteria, and have a CLL IPI score of 4 or higher and or complex cytogenetics, which we define on this study as 3 or more cytogenetic abnormalities. CLL IPI score is, of course, a combination of stage, age, status of their IGHV gene, beta-2 microglobulin, and presence of deletion 17p or TP53. These patients are randomized 2-to-1 to early intervention versus delayed intervention with venetoclax and obinutuzumab given per standard of care and as it was given on the CLL-14 study, so 6 months of obinutuzumab therapy and 12 months of venetoclax therapy. All of the drugs are provided in this study, whether the patient is on early or delayed intervention, so it does come with a financial advantage to patients for enrollment. Primary endpoint of the study is overall survival, and we need a total of 247 patients to achieve this goal. Secondary endpoints are listed here, but include safety, response rates, times to next treatment. Translational endpoints, the big one is looking at MRD status and development of resistance to therapy. So a brief update, this study did open as of December 2020. We currently have 18 out of 247 patients accrued. You can see the high accruing sites. There are quite a few from the SWOG group. So thank you for your contributions to this study. Just highlighting University of Rochester, Cleveland Clinic for accruing some patients. I think there is one patient from City of Hope now on study as well. We are undergoing an amendment that, just so you know, and so your coordinators know, there was an error as to where the minimal residual disease testing should be sent. So just that is updated now in the current version, but just highlight that so that no one uses an old version. Of course, on this study, I think, Evusheld coming into the clinics has been a big advantage because a lot of folks had been nervous during the height of the pandemic enrolling patients that might receive extra therapy. But now that Evusheld is available, I'm hoping that that might make people feel a little bit more comfortable enrolling these patients. But again, I would really love if any of you don't have this study open to help out with accrual to this study. And, just keep in mind, like I said, these patients have to have their risk factors checked early in the course of disease to get them on the study.
Great, thank you. I do think with Evusheld, it should get a little bit better, so. Paul?
Yeah, trying to look for these patients, I think it's a little bit challenging, but I think it really takes a shift in mindset as you're meeting new CLL patients. You obviously have to really look at the CLL IPI score with every patient diagnosed in the last 12 months. And if someone has deletion 17p, it's an easy way to get them into the study, but it's not often that we think, okay, I have an older patient, they have palpable lymphadenopathy. If they're IGHV unmutated, that's another way to get the CLL IPI of 4. So it really takes that thought process to look for these patients, which isn't something we often do. So something we have to think about, educating people to sort of go through that process as they open the study at their site, and checking in with some of those PIs if they see any other barriers, just because obviously it's an important study and it's, the accrual goal is lofty. So yeah, something for us to think about.
Yeah, thanks, Paul. And there was some data published that looked at the rate of just standardly people doing these tests on CLL patients at diagnosis, and it was pretty low. I mean, it was less than 15%. And so hopefully that study has got out and, kind of helped to show people that this actually can be helpful. Just even if we're not enrolling on this study, it helps with prognosis and it helps a patient understand, what their course of disease could look like.
Liz is making the point on the chat about beta 2 as well, which may not be in everybody's standard workflow.
But I think, both of you are highlighting just a huge educational gap for providers. And, maybe there is something we could do, to think about addressing that.
Hildy, do you have, both from a patient's and provider standpoint, I know on a couple of the other trials, we've put together some special materials. This is really Dr. Erba's job, but I'll step in and just ask.
Yeah, thank you. I do think that, and it may be too soon to really tease this out, but in addition to COVID, and we had talked about this early on when you were first developing this, one of the the issues with CLL is the tradition of telling people you don't have to be treated now. And now to be thrown into a trial where it's not known whether starting therapy now or delaying it is going to make a difference. In addition to being in the age of COVID, people are more likely to say, I'll wait if I don't have to have treatment. And I don't know if you're getting that feedback, but it would be great because it's another, if we're going to be doing some education for providers, it would be a great opportunity in terms of shared decision-making to really tease that out and explain to them that, this is answering a very important question and may actually help them. So I think that there's a lot of education that needs to be done. And also for that, the nonprofit organizations do a lot of education around this for the patients, but hard discussions with patients.
Yeah, no, those are really great points. And I do think the most success I've had in enrolling these patients is that the discussion starts as soon as I meet them, at their diagnosis. And I say, at the current time, standard of care is observation, but we're going to do some additional testing that might indicate you're at a higher risk. So we'll have a different option for you if in the case that you do have high risk.
Great.
Thanks.
Thank you. Okay. So I just want to thank everybody for being here at this meeting, and also those who are on Zoom. This is a really incredible and full agenda. I'm really proud of all the hard work that's been done, and really want to thank everyone who's opened the trials. Continue to accrue. There's meaningful progress here, and really excited to be a part of it. I want to thank all of our speakers for some really outstanding presentations and concepts that are in development, and invite any feedback after the program as well. Anything you want to say?
Echo all of those remarks. And if you have feedback for us on ways we can do better at this committee meeting for moving forward, just reach out. We're happy to take that advice. I think the plenary is at 12, so this lets you get a little snack before then.
Great. Thank you. Have a great rest of the day.
Excellent. Safe travels.
We skipped this last trial here. It wasn't in the book. Oh, well.
That was the econ.
Yeah. 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 I don't know why I didn't make it to the.
That was my fault.
Oh.
Probably. So I also otherwise somebody else.